Okay, good evening. Can you hear me? Seems that I'm lagging. Hello? Hello, can you hear me? Hello? Okay, good evening. Can you hear me? I'm having problems, technical problems. But can you shout out in the chat box if you can hear me? I think I'm suffering a little lag. Okay, thank you very much, Jocelyn. Okay. Can you hear from the others? But I have audio. I have audio. But I think I'm lagging. But you can hear me, Jocelyn. My audio is okay. Okay, and anyway, good evening, everyone. Welcome to Biochem Serie Live. I'm happy that you dropped by, and I would like to introduce myself. I'm Doc Bao. I used to teach biochemistry for 25 years, and I reactivated this channel. This channel aims to teach biochemistry and make difficult topics a little easier. Okay, so for tonight, I have scheduled chemistry of coenzymes. Okay. Sige. But I'm on, right? I'm on. Okay. So for coenzymes, there are 11, and I'd like to welcome the viewers now. Sorry for the technical difficulties. As they say, it's Murphy's law. No? If something can go wrong, it will, and it did. Okay. But you can see me. Okay. Anyway, so good evening. So this is your chemistry of coenzymes. I would like to welcome everyone. I go to coenzymes, I would like to revisit the concept of enzymes. You know very well that enzymes accelerate reactions by lowering the activation of energy, and the enzyme by itself will not. It will change transiently, but eventually it will go back to its former, okay, to its former form, and you will now produce your products from your substrates. Okay, but your enzymes cannot do it by themselves. They need helpers. Okay, so these helpers are your coenzymes. Okay, there's a little problem with semantics, a little confusion, no? Because we're more familiar with the word coenzymes, right? But a more all-encompassing term is your cofactor. Okay, so your cofactor is non-protein portion of the enzyme. It usually there are small molecules, or sometimes there are even metallic ions, but they are needed by the enzyme to function. Okay, so this is the algo, no? This is the schema. When we say of holoenzyme, this contains your apoenzyme. Your apoenzyme is your what contains your apoprotein. So it's your protein portion of your enzyme. Enzyme without your cofactor or without your non-protein portion, this is your you call this your apoenzyme. Okay. Sige. The others are just coming in. Okay. So I think a better term is your cofactor. No, cofactor is more inclusive because your cofactor includes your metal ions. No, if your non-protein portion of your holoenzyme is inorganic, no, they're just plain metallic ions. Okay, and if your non-protein portion may be organic, and this is under your cofactor. If your non-protein portion or your cofactor is organic, okay, we call that your coenzyme. And we will see, no, it's further dichotomized into two, right? So your inorganic metals, if it's tightly bound to your enzyme, it is your metal enzyme. But if it is not, no, it's just there to stabilize. For example, the negative charges of your phosphate groups in your DNA, they are only your ion activators. Okay, so if we're going to visualize it in an image, now this is your apoenzyme. plus your cofactor. So cofactor is much more inclusive than your coenzyme. This is now your holoenzyme, which is active. Your apoenzyme without your cofactor is inactive. Okay, so your cofactor is under your cofactor. Your coenzyme is under your cofactor. So your coenzymes, as I've said, they're small organic molecules, and most of them, 8 out of the 11, are derived from your vitamins. No, so right out of our mind, we don't know no, what are the, if there are 11 and only 8 are derived from your vitamins, three are derived from something else, okay, from an acid, etc. I'll tell you how to remember later, okay? So they are modified during the reaction. For example, NAD, that's the active form, okay, it becomes NADH. In order to go back, no, to its oxidized form, it needs another reaction. Okay, it needs another reaction or another enzyme catalyze reaction to restore them to their original state. Okay, and unlike enzymes, enzymes have specific, they have specificity. Okay, your coenzymes, it does not follow that they're specific to one reaction only. No, sometimes they have varying substrate specificity. A lot of them have multiple, okay, substrate specificity, but some is unique only to one. Can I have a shout out? Is my audio and my video okay? Kasi from my end, from my StreamYard point of view, para I'm stopping it. But you can hear me. Can you hear me? Can you give a shout out? I fixed the settings, no, so that once you type, I can in 1 or 2 seconds I can see your okay, my audio is good. Thank you. Okay, so thank you. Okay, mas delay naman ngayon yung StreamYard. I'll just look into the YouTube. Okay, so shout out to Angel. Thank you very much. To JMS Bondok, to Thea Gulay, to Jesly Anton. Audio is fine. Thank you very much. Okay, so sige, ano nga natin, no? Ipakita natin sa ano. So we'll show this to you. Okay, so thank you, Jesly. Ha, sige, I'll go back to the ano. But I'm okay. I'm okay because I cannot see your reaction. But feel free to drop by in our live chat box. Okay, sige. So you have 8 out of 11, and here, no. So your cofactor, okay, or your organic portion is further subdivided into two. If the coenzyme is tightly bound, okay, most of them are tightly bound, not to your enzyme. Now they're part of the enzyme, but they're not inherently the enzyme itself. Now they're bound a lot. No? For example, your FAD, etc., your lipoamide, they're tightly bound. They're called your prosthetic group. As you can see here, to start with, no, they're already bound to the enzyme, and they don't leave. However, okay, if your coenzyme is loosely, no, it's not bound, but will just participate, or will participate in the reaction, then eventually go away. Um you call that your co-substrate. You call that your co-substrate. Okay, so as I mentioned, 8 out of the 11 are derived from your vitamins. So we will go to this each one later on, ha. So children, mga anak, medyo marami to kasi we have 11, but I shifted through all the fluff. No, I remove all the fluff and will just concentrate on the must knows, not the usual ask questions. Okay, so you have the different vitamins. Now the different B vitamins will go to each one, and you have the different deficiency states. No, very for B1, B2, etc. No, B3, your pellagra, your pernicious anemia for B12. If you're going to look at this right out of your mind, no, top of mind, can you tell me what are the three coenzymes that are not vitamin derived? What are the three coenzymes that are not derived from vitamins? Ngayon mahirap, right? It's hard, but just remember your TLQ, okay? Two lovers quarrel. Okay, so two lovers are quarreling because they're quarreling because they don't have vitamins anymore. Pinag-aawayan nila yung centrum. Pinag-aawayan nila yung vitamins, okay? So your two lovers quarrel, TLQ. Okay? Okay, so Rhea, right, not TLQ, no? This is T for tetrahydrobiopterin. Tetrahydrobiopterin L for lipoic acid, or we will see later on it's better termed as lipoamide, okay, because you don't have any free lipoic acid. And Q, simple, very, very easy to remember, is your ubiquinone or Q. So you have 11. You have 11. You have 8 that are derived from your vitamins. We'll go to those later on. And you have your 2 lovers quarrel. So in one snap, you can remember. Okay? Sige. So this is our coenzymes info shelf. What I did is on your left side, no, on your left side, I hope you're using a big monitor, no, but in your iPhone, maybe in your iPad, you can see that. What we're going to do is we're going to classify. You have 3 classifications of your coenzymes. You have coenzymes that participate in your oxidation reduction. You have 4. Okay. You have coenzymes that are that does not only transfer, okay, functional groups, but will activate them as well. And lastly, you have 3 coenzymes that will just transfer them. Then each of the 11 coenzymes, we will go through this. Okay, we will classify them. That's the first. We will study them, not to put order. Okay, to put order and organization in this discussion, we will discuss them in their proper order. Now we'll go through the 4 oxidation reduction coenzymes first. Then we're going to the activation transfer coenzymes second. And lastly, now you have 4 as well for your activation transfer coenzymes. And you have your transfer coenzymes that are 3. So these are the things, not that I'll write them on the left. No, I'll actually, you'll see them right outright. Okay. So the classification from which vitamins they are sourced from, and if they are not sourced from your vitamins, we will see where they are sourced from. No, what are their sources? Are they sourced from an acid, etc.? Then very, very important, the chemistry. No, chemistry is a study of structure, and once you can remember the structure. No, you don't need to memorize and write it from your mind. Okay, we just have to maybe to recognize, okay, or to remember salient features of their chemistry, so that will tell us a little about their properties, and that will tell us about a little about their their function as well. And very, very important, now what is the active site? The active site, some books will tell this as your functional group, no, functional site that will do most of the heavy lifting. Okay. Then next is will see their function. No, very, very important. No, do they transfer electrons? Okay, do they remove carboxyl groups, or do they add on carbon dioxide, etc.? Then we will go to examples. No, I look up to all the lists. Okay, I will list all the reactions that are facilitated by the reactions that these coenzymes will help to completion. Okay, and others. Now, if there are any nuances, for example, one is inhibited by raw fish, etc., or one is responsible if there's deficiencies for pernicious anemia. Okay, I'll put this under. No? I'll put this under others. Okay, so for your redox coenzymes, we have 4. We have 4. No, you have your pyridine nucleotides, your flavin nucleotides, your coenzyme Q, and tetrahydrobiopterin. You have 4. Okay, and it's worthwhile. No, it's prudent to know that what they do is they participate in the transfer. No, they get and they release your electrons. No, you know that oxidation basically is the loss of electrons, and reduction is the gain of electrons. No, one is an oxidizing agent, one's a reducing agent, etc. No, so what these coenzymes will do is they will receive, they will fetch the electrons, and they will release it to other substances. Okay. Then activation transfer coenzymes, you have 4 as well. No, you have thiamine pyrophosphate. No, you have pyridoxal phosphate, your coenzyme A, and your biocytin. Then your transfer coenzymes, you have 3. No, you have your lipoic acid, your tetrahydrofolic acid, and your cobamide coenzyme. I know, children, no, there are a lot. Okay, there are a lot. So just bear with me. No, I'll try to make this as fast as possible. Then at the end, no, I prepared some questions. So I look at certain books, no, they have questions, and I looked at former exams, the usual questions, and I did try to answer the questions, and they're confusing. Okay, so what I suggest and what I recommend is when you study this. No, I know, I think you have your exam on most of you will have your exam on Wednesday, then you're shifting exams in a week after. I suggest that you study the coenzymes at different times. Okay, you put a temporal distance. don't study them. Don't cram all 11 in one sitting. No, for example, your redox coenzymes you study this Sunday morning, then your activation transfer coenzyme at night, so that you have a temporal boundary. Okay, so let's now start. Now your oxidation reduction coenzymes, you have four, and as I mentioned, they participate in the transfer of electrons. Now this is just a brief. This is just a review. Oxidation is the loss of electrons. Now when you're oxidized, you lose electrons. That makes you a reducing agent because you can contribute the reducing equivalents. Okay, your reducing equivalents, and usually they come in pairs. Okay, they come in pairs. You'll see later on. And if you're an oxidizing agent, you're the one that is reduced, no, because you receive the electrons. Okay, the enzymes that will facilitate or catalyze these reactions are your oxidoreductases, right? But they need their helpers. They need the moieties that will accept the electrons and that will keep them and that will release them. Okay, so if a molecule is oxidized, I've mentioned, no, it loses electrons. A good gauge, a good clue. Okay, a good clue is if the products have lesser number of hydrogen, okay, or gained an oxygen, that is oxidation. Okay, that is oxidized. Because your electrons, you know very well, they are in your hydrogen atoms. If you lose a hydrogen atom, you lose your electrons, you are oxidized, but you are a reducing agent. Okay, but if you gain hydrogen, okay, or you lose an oxygen, that is reduction. Okay, no. So the first two that we'll study will be your nicotinamide, your pyridine nucleotides, and your flavin nucleotides. Okay, they can transfer your electrons. So let's now start, no, with your pyridine nucleotides. They are pyridine nucleotides because they are your nucleotides. You have your pyridine, no, your pyridine nucleotides. Okay, and they are from your niacin or nicotinic acid. Niacin or nicotinic acid, doc, vitamin B3, no? Sometimes it's really very confusing. Okay, remembering the names. But if you just mention niacin, no, niacin, you have three, okay, you have three syllables, so that's vitamin B3. Okay, niacin is vitamin B3. It might be funny, but I hope that can help you. Okay, niacin, that is vitamin B3. You have your nicotinamide ring. Okay, you have your nicotinamide ring and you have your AMP. You have your adenosine monophosphate. Okay, so sometimes it's your dinucleotide because you have your two nucleotides that are joined together, no, by your pyrophosphate bridge. They're found. The niacin is found in fish and meats, fortified foods, cereal and bread, and in legumes. And if you're deficient, no, we'll not go into the mechanism, no, because it goes to tryptophan, etc. Quite a long process. Just remember, no, if you're deficient in niacin, that is your pellagra. No, it's here. Okay. So this left side, these are the most important things. Okay. So what you can do is, if you have time, no, you can take a picture or you can print this out. No, you can expand this, and I hope it can be seen naman, no, hopefully in your screens. Okay. So that's niacin. That's vitamin B3. So it's dinucleotide because you have two nucleotides, now two nucleotides that is joined by by their phosphate groups by a phosphoanhydride bond. So that is your dinucleotide. Now, dinucleotide, two nucleotides. The AMP is derived from your ATP, okay, and your nicotinamide is a pyridine derivative. So that's why they are called your pyridine nucleotides. But they are not nucleotides just like your ATP or GTP. They are not nucleotides in the truest sense of the word. So they are pseudonucleotides. They are pseudonucleotides. We're now in your chemistry, okay? They are pseudonucleotides. And they are bound. So please remember, but it's easier, no? It's easier to remember that most of your coenzymes are prosthetic groups. So they're bound, okay? They're bound tightly to the enzyme. Most of them are bound tightly to the enzyme. I think we have only one or two exceptions, so we'll see later on. So what is the active site? The active site is the C4, no? The C4, okay? The C4 of your nicotinamide ring. That is opposite to your nitrogen. No? This is positive. So what happens is that it 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 more or less gets the electrons. So this makes your hydrogen acidic. Okay, this makes your hydrogen acidic. So because it is because it is partially positive, no, it will now accept the hydride ion. So guys, no, when you see words that are in red, they're very very important. Okay, they're very very important. I think I mentioned this when I discussed your bioenergetics. Your hydride ion is what? What is your hydride ion? Anyone? If you remember, your hydride ion, sometimes this most of the time this usually asked. Okay, your hydride ion is your hydrogen atom plus an additional electron. A hydrogen atom plus an additional electron. Okay, so it is negatively charged. So from here we can see that you have your twin. No, you have your two reducing equivalents already that is incorporated in one hydrogen atom. And where do we see your NAD? Your NAD, you see that in complex one, right? In your NAD H dehydrogenase. Right. So please, no. So this is your C4. Okay, that is your C4 of your nicotinamide ring. So those that are very very important are on the left side. Okay, so it attracts the hydride ion and you form your quinonoid. Okay, your quinonoid is a six-membered, okay, six-membered heterocyclic ring. No, because you have nitrogen and you have two double bonds. Now, when you see your quinonoid, no, parang it sounds funny, right? But that's a name, no? Quinonoid, you have two double bonds. Okay, you have two double bonds. So from your nicotinamide ring, you add your hydrogen ion, your hydride ion, sorry, no, your hydride ion. So the substrate that were in your hydride ion got is derived, no? So that substrate is oxidized, will give two electrons in one hydrogen, okay? In one hydrogen. Where is the other hydrogen go? It will go into the solution. Kaya usually when you see NADH, you have NADH plus H, right? NADH plus H because the H goes into the solution. So that's that. No, you have your quinonoid structure. So your NAD, now this will what? This is your oxidizing agent. Now it will accept the electron. This will get reduced. This is reduction. So NAD is the oxidized form. Your NADH plus H, that is your reduced form. So most of the time, your NAD, now when you see NADH, this is for oxidation reactions. Oxidation reactions. Substrates will lose their electrons and your NAD positive, the oxidized form, will get the electrons. Just like in complex one, right? Your NADH will bring the electrons in the form of your hydride ions, right? Okay. So usually this is for your oxidation reactions. On the other hand, the NADPH, no, we don't see this much, no, but you will see this. You will see them in your fatty acids by synthesis. You will see them in your hexose monophosphate shunt, no, in your pentose phosphate pathway. Okay, your NADPH is used for reductive synthesis. No, they will now be the sources of your electrons for synthesis. Okay, for synthesis. So take note of this. Okay, so so these are the reactions. Okay, so these are the reactions that are commonly facilitated by your NAD. No, your lactate dehydrogenase. Okay, your malate dehydrogenase, your pyruvate dehydrogenase. Okay, your oxidation of your lactate to pyruvate, malate, etc. Pyruvate dehydrogenase complex. We will see more of this later on. However, your NAD positive, no, NADPH, this is for your reductive, okay, reductive synthesis. You'll see this in your glucose 6-phosphate dehydrogenase. You see this in your hexose monophosphate shunt, and you will see this in your fatty acid synthesis. So these are the reactions that are catalyzed. Okay, and more important thing is, sorry, no, it is usually no, your oxidized form that will absorb. Okay, your oxidized form that will absorb a light at a certain wavelength. Now you will see later on of certain coenzymes that will exhibit maximum absorbance at a certain wavelength. So that's the first. Okay, so what are the special things that we have to remember about your NAD with your pyridine nucleotides? The active site is at C4. It transfers hydrogen. It transfers electrons in the form of hydride. I forgot to put it here. But please remember, they accept and they release electrons in the form of hydride. The next redox are your Flavin nucleotides. Your Flavin nucleotides. Your Flavin nucleotides, yan. This is now sourced from your vitamin B2, no, siguro Flavin. But I'm sure you know that riboflavin is vitamin B2. There's no confusion over that, no. But those that are not familiar will be your B7, your B9, etc. Now we'll go to them later on. So they're synthesized from riboflavin. Not this, you don't really have to remember this. No, you just remember that they are from your riboflavin. Then you have your flavin adenine dinucleotide. Okay, so your FMN, you have a single, okay? You have a single nucleotide, but if you have two nucleotides, not it's your flavin adenine dinucleotide. So both of them contains your isoalloxacin ring, okay? Your isoalloxacin ring and your sugar ribitol. Your isoalloxacin ring and your sugar ribitol. So it is not a true nucleotide as well. So it is also a pseudonucleotide. So what are the two pseudonucleotides that we have studied? The first two. Okay, your pyridine nucleotides, your NAD, NADP, okay, they are what? They are pseudonucleotide, and your flavin nucleotides are pseudonucleotides as well. So please remember that this is the one that contains your ribitol. Okay, so what's the active site? The active site is your N1 and N5 of your isoalloxacin ring. Your N1 and your N5. We can see here that you have two sites, right? You have two sites because flavin nucleotides, your FAD and your FMN, will receive or will release electrons in the form of hydrogen atoms. In the form of hydrogen atoms. So they accept a maximum of 2 electrons and 2 protons. You know very well the hydrogen atom is an electron and a proton, right? A hydrogen atom is what? No, it has a proton and it has an electron. So if you lose a hydrogen, you lose an electron and a proton. Sometimes no oxidation is also known as dehydrogenation. Dehydrogenation, no, you remove your hydrogen. Okay, you remove your hydrogen, so you lose electrons in the form of hydrogen atoms. So FAD undergoes reduction in 2 steps, okay, because you have two sites, no, the N1 and the N5 of your isoalloxacin ring. Please do remember the functional group, okay, or the active site. In this case, it's N1 and 5, no, for your NAD and NADPH, it's your C4 of your nicotinamide ring. Those are the things that you have to remember. I know there are a lot, but we cannot escape them. So what is the function? No, the function is what? No, they're redox coenzymes. So they will just accept 1 or 2 electrons. Okay, 1 or 2 electrons, 1 or 2 electrons. So if you start with the fully oxidized flavin, no, this is your fully oxidized flavin, na your quinone, if you take in 1 electron, it becomes your semiquinone. Semiquinone, okay? It has unpaired electrons, kaya nga this can be a source of your reactive oxygen species eventually. Okay, your reactive oxygen species. However, no, in order to protect us, oh, God is so great, okay, they make your FAD tightly bound as well, no, just like your NAD, no, they're tightly bound to your enzyme. So they will now release or accept 2 electrons in the form of 2 hydrogen atoms. Okay. So kaya if you can see here, no, your fully reduced flavin, no, this is your quinol. Your reduced flavin has this formula, no, FADH2 or FMNH2, no, FADH2 or FMNH2, because your electrons are what, no, are contained in 2 hydrogen atoms. 2 hydrogen atoms. Okay. So transfers electrons and protons during redox reactions in the form of hydrogen atoms, 2 hydrogen atoms. Your NAD, your NADPH contained in 1 hydride ion. Okay, 1 hydride ion. So the reactions that are catalyzed, I'm sure you're familiar with this. Now, this is your complex 2. This is your complex 2. The oxidation of succinate to fumarate is what? Facilitated by your FAD, by FAD. No? So it will what? No? Succinate will lose 2 electrons in the form of 2 hydrogen atoms. 2 hydrogen atoms, and eventually it will be transferred by the next coenzyme, no? Your ubiquinone. So these are the reactions catalyzed. Okay, oxidation of succinate to fumarate by your succinate dehydrogenase. Okay, we learned that, no, this is in your complex 2 of your electron transport chain. Complex 2 of your electron transport chain. It is also involved in your beta oxidation, no, yung beta oxidation of your fatty acid. No, Doc, I know we're not yet in metabolism, right? That is in your second shift, I believe. Okay, but you will meet your FADH2, right? Okay, if you remember, how many ATPs will be produced if you have a mole of your FADH2 entering complex 2? Now this is the reason, no, because it bypasses your complex 1, etc. But that's another topic. Then you have your L amino acid oxidase, no, just in case, no, but just please do remember succinate to fumarate and your beta oxidation, no, your FAD. Okay, your FAD participates in that. Okay. Your FMN, that's the reason why I put this here. No, maybe you might be curious, Doc. What about FMN? No, FMN will facilitate your L-amino acid oxidase. No, this is in your amino acid metabolism. Amino acid metabolism. So this is one of the nuances. No, this is one of the other properties. As I mentioned, it is the oxidized form. Okay, it is the oxidized form that is intensely colored and that will absorb maximally at 450 nanometers. 450 nanometers. But once it is reduced, no, it will be colorless. Okay. Sometimes this is used in measuring the activity of your of your coenzymes or even the enzymes, no, that they are facilitating. Okay. Next is your ubiquinone. Ubiquinone. I'm sure you're familiar with the first three, right? Because they are in your electron transport chain. No, they're part of your universal electron acceptors, right? NAD, NADH that is in complex one, FADH2, no, succinate dehydrogenase that is in complex two, right? And you know that your ubiquinone will accept the electrons from your NADH from complex 1 and from your FADH2 from complex 2. And what will accept this with open arms? It is your ubiquinone. Okay, it is your ubiquinone. Is my audio still okay or is my video, am I choppy? Maybe it's just from my end. Can I have a shout out from anyone? We have 16, no? We have 16 viewers. Thank you very much for dropping by. Okay. Anyway, I'm making this fast so that we can reach the questions. Hopefully, we're still all around. Okay, thank you, Thea. Thank you, Thea. Thank you for listening. If you have any comments or if you have any questions, you feel free to chop in. Thank you. Okay. Now it's different, no? From StreamYard. What is now faster is from YouTube. No, audio is fine. Good. Thank you, Angel. Thank you very much. No, I'm happy that everything's holding up. Okay, let's now go to your ubiquinone. What are the three, again, what are the three coenzymes that are not sourced from vitamins? So this is the first one that is not sourced from any vitamins. You have your TLQ. No, you have your TLQ. You have your two lover squirrels. Okay, so you have tetrahydrobiopterin, your lipoamide, lipoic acid, and your ubiquinone. No, that's Q for quarrel. That's two for quarrel. Okay, so TLQ. So from this is not sourced from vitamin, but this is a good, nice to know. No, it is sourced from your tyrosine. Okay, it is sourced from your. You have other sources as well, no, but your ubiquinone can be synthesized, no, and sourced from your tyrosine. So it is not considered the vitamins. It's mammals, no, and in humans we can synthesize it from essential amino acid tyrosine through an 8-step process. Now it might be tedious, it might be you have a lot of steps, but it can still be what? It can still be synthesized. And why is it known as ubiquinone? Why is it called ubiquinone? Because it is ubiquitous. Now, ubiquitous meaning it's everywhere. Okay, it's everywhere. It's in a lot of your organisms as well. Okay, but we know very well that in your powerhouse of the cell, no, your mitochondria, ubiquinone is really very important. So what is the structure? No, the structure is you have a benzoquinone. Okay, you have a benzoquinone. That's why it is called Q. Okay, coenzyme Q or ubiquinone because of your benzoquinone group. No, you have a quinone group. You have a quinone group with a very long isoprenoid side chain. No, you have 6 to 10 units. Okay, isoprenoid. No, so in higher organisms like humans, the number of isoprenoid side chains and number of isoprenoid units is around 6 to 10. But this is very, very long. What property will it get if it has a very, very, very long hydrocarbon chain? Around 50 carbons. No, that's long. Around 50 carbons. No, 6 to 10, around 50 carbons. That's long. This will now confer to it hydrophobicity. Okay, hydrophobicity. So it is small, it is mobile, it is not protein bound. So if there's a question, which of the following coenzymes is not firmly bound to the enzyme? Your ubiquinone, that is your ubiquinone, because it is mobile. You remember, no, in your electron transport chain, it collects the electrons from complex 1 and complex 2, right? And it brings it to complex 3. No, that is your ubiquinone. And it is also a redox coenzyme because it carries and transfers your electrons. And it is diffusible within the lipid bilayer because of your isoprenoid side chain. Okay? Because of the long isoprenoid side chain. So what is the active site? No, the active site here is the carbon 1 and carbon 4. Carbon 1 and carbon 4 of the benzoquinone ring. No, it's similar, no? Parang it's similar to your isoalloxacin ring, no? You have your quinone form and you have your quinol form, no? Yung quinol form, and you have a semiquinone form as well. So in your mitochondria, okay, your rapidly respiring mitochondria is a source of your reactive oxygen species. I'm not sure if you already have your lecture in your oxygen metabolism and toxicity. Have you had that? Your oxygen metabolism and toxicity, the reduction of oxygen, right? But sometimes now you release your reactive oxygen species, no, your reactive oxygen species that can cause oxidative damage. Okay, thank you, Canteroni. No, so thank you very much. No, so this is the reason why. No, if sometimes you escape, no, the electrons escape and can cause what a chain reaction, no, and produce your reactive oxygen species. So in this case, no, your coenzyme Q, just like your flavin, no, they will what? They will transfer electrons in the form of hydrogen atoms as well, in the form of hydrogen atoms. So this is your fully oxidized. Now this is your quinone form. It takes in one electron in the form of hydrogen. This is your semiquinone. Now you have an unpaired electron, and this makes it dangerous. Okay, it makes it dangerous, and this is not strongly bound, right? It is not bound to your enzyme. So this is the source, no, of your superoxide, no, because the lone pair of electrons, no, can cause a single reduction of an oxygen molecule producing your superoxide, right? Okay, what's needed really is a second electron to fully reduce your semiquinone to your quinol, okay, to your quinol form. If this is Q minus with a dot, and you have your QH2. You have your QH2. Okay, so these are your active sites, no? Your C1 and C4 of your benzoquinone ring. And you have a stepwise transfer of electrons and protons in the form of your hydrogen atoms. Okay, in the form of your hydrogen atoms. Function, same, no? Just like the first two, just like your NADH and your FADH2, it can transfer electrons. Okay, so because it can act at a junction between one or two electrons that can be accepted, and it is lipid soluble and it is mobile, no, it really plays a very important role in your electron transport chain. And again, please remember, the electrons are transferred in the form of hydrogen atoms. NADH, NADPH in the form of hydride ions, no, FADH2, FMNH2 in the form of hydrogen ions, so ubiquinone as well in the form of the hydrogen atoms. So the reactions catalyzed is I think this is the only, okay, this is the only function. This is the only function of your ubiquinone, your coenzyme Q or your Q. No, it accepts, no, it receives the electrons that are brought by your NADH, no, from a lot of your reactions, okay, from a lot of your reactions in the in inside the matrix. For example, your TCA, no, your tricarboxylic acid cycle, etc., your beta oxidation, right? So it brings the electrons in your in the inner mitochondrial membrane. Then in your complex 2, no, you have your oxidation of succinate to fumarate. No, the two electrons are released, and which will accept this will be your coenzyme Q or your ubiquinone, right? So that's the main role. And I know, no, I'm sure, I hope you remember your PO ratio, right? Why you have a lot more ATP yield, no, from NADH rather than from FADH2. FADH2 will only have what, no, 1.5 or 2 because it bypasses your complex 1, right? I hope you do remember that. Then especially an additional information. No, it is the oxidized quinonoid. No, that's the quinonoid again. No, quinonoid. You have a six-membered ring with two double bonds. Okay, that's quinonoid that will absorb at a certain nanometer. Don't you don't have to remember. I believe you don't have to remember the wavelength. I you just please do remember which can absorb and which form. Okay, that's important. It's best that to remember that it's almost always the oxidized form, no, that will accept that will absorb maximally at a certain wavelength. Okay, so we're progressing very well. Huh? So the last is your tetrahydrobiopterin, your BH4. This is also a redox coenzyme. Okay, it is again, it is not one of the it's not one of the coenzymes that are sourced from your vitamins. So this is the T in your TLQ. Okay, that is your tetrahydrobiopterin, and please do remember it is from your GTP. No, your biopterin is synthesized from your GTP, your guanosine triphosphate. So if there's a question, where is your BH4, no, your BH4 synthesized and you don't find guanosine triphosphate, the answer might be in your biopterin, okay? The biopterin. So it is synthesized from your guanosine triphosphate by several steps. It's not important to remember the steps or the enzymes. Just remember that the source of your tetrahydrobiopterin is your GTP, your guanosine triphosphate via your biopterin. No? Your biopterin is synthesized via guanosine triphosphate. So the chemistry is that it is there's not much information. I keep on looking and looking what's the special chemistry, no? But what's mentioned in some books is also known as your sapropterin. Sapropterin, it's a substituted tetrahydrobiopterin, okay? Substituted tetrahydropterin. So it's a reduced pteridine derivative. There's a quinonoid. It's a quinonoid dihydrobiopterin. Okay, so that's that. So just I hope we can more or less recognize it. So what's the active site? No, the active site is what? No, the nitrogen in your what? No, in your biopterin ring. The nitrogen in your biopterin ring. So what do we see? No, we can see that it can accept electrons in the form of hydrogen atoms as well. Okay, so it transfers electrons and protons, but a very important caveat, a very important qualifier is that it is what involved in hydroxylation. So it does not only involve itself in transferring electrons, but it is also involved in aromatic ring hydroxylation. I think I have a yellow. Now, if you can see, the yellow boxes, I hope of these are just special memory aids. Tetrahydrobiopterin, okay, so that is BH4, no, hydroxylation, no, because me personally, I find it really hard not to remember all of these different things in the 11. They're not only five. Okay, you have a lot of things to remember. Sometimes when they're all together, bunch up in a series of questions, it really gets very, very confusing. Okay, so your tetrahydrobiopterin is important in hydroxylation of your aromatic amino acids. If do you remember, no, in your amino acid chemistry, what are the aromatic amino acids? No, how many aromatic amino acids do we remember? First of all, na top of mind, you have your phenylalanine. No? If you hydroxylate this becomes your tyrosine, right? This becomes your tyrosine, and one hydroxylated amino acid as well that is double ring, that is your tryptophan, aromatic, no, amino acid, that is your tryptophan as well. So from here you can see that your phenylalanine is converted to tyrosine via your phenylalanine hydroxylase. Your tryptophan is converted to your 5-hydroxytryptophan by your hydroxylase, etc. No? So all of these hydroxylases will need your tetrahydrobiopterin. Okay, your tetrahydrobiopterin. So transfers electrons and protons for hydroxylation reactions of aromatic amino acids. And we will see later on, no? You have a lecture on nitric oxide, your nitric oxide, chemistry of your nitric oxide. Okay, so please do remember this is also the coenzyme that is involved in your nitric oxide synthesis. I'll show you a reaction later on. Okay, So you have your NOS, your nitric oxide synthases, no, your NOS1, NOS2, NOS3. All of these nitric oxide synthesis will also what no will take into the part take into it no the participation of your tetrahydrobiopterin. Sige, if we're going to look at this, no, this is the classical example, okay, of a hydroxylation reaction. Phenylalanine is a essential amino acid. It's one of the most hydrophobic amino acids. It is essential. Your tyrosine, your tyrosine is partially acidic, right? It is slightly acidic and it is not essential. Why is tyrosine non-essential? It is non-essential because you just simply hydroxylate your phenylalanine. You hydroxylate phenylalanine and boom, you have your tyrosine. No, you need your phenylalanine hydroxylase. Phenylalanine hydroxylase is a mixed function oxidase. No, it's a mixed function oxidase. It is an hydroxylase. What is a mixed function oxidase? No, oxidase because it incorporates oxygen. Okay. So this is oxidized, right? This is oxidized because you incorporate your oxygen. It is mixed function because in molecular oxygen, you have two oxygen atoms, right? You have two oxygen atoms. One of the oxygen atom in your molecular oxygen is incorporated into the substrate. It's incorporated into phenylalanine. And one of the oxygen is incorporated in water. No, that is your mixed function oxidase. No, so your phenylalanine hydroxylase, okay, your tryptophan hydroxylase, your tyrosine hydroxylase, they are mixed function oxidases. And what is important no, in our discussion is that it what no it uses your tetrahydrobiopterin. This is tetrahydrobiopterin. This is dihydrobiopterin. No, this is the fully reduced. This is the fully reduced. No, it contains the electrons, right? It is oxidized to your dihydrobiopterin. Okay, so this is the oxidized. The problem here is this has to be regenerated back to your tetrahydrobiopterin by your dihydropteridine reductase. No, your dihydropteridine reductase, your DHFR. Okay, so that will now regenerate your tetrahydrobiopterin. This is the enzyme that is what no prevalent in your classical phenyl phenylketonuria. Okay, phenylketonuria. Okay, if your phenylalanine hydroxylase is there's something wrong, no, it's deficient, it's defective, or it's absent. What happens? Your phenylalanine, what? Your phenylalanine accumulates, right? And you don't produce your tyrosine, right? Your tyrosine becomes essential. That is your classical. However, if you have a problem in the synthesis of your tetrahydrobiopterin from GTP, or if your dihydropteridine reductase is deficient, you don't regenerate your tetrahydrobiopterin. That is your PKU type 2, type 3, etc. The rarer forms of your phenylketonuria. So from here you will see, from here you will see, I forgot to mention that your tetrahydrobiopterin is important in the synthesis of your neurotransmitters, like serotonin, melatonin, dopamine, and your catecholamines, norepinephrine, and epinephrine. So if you see these words, you automatically remember your phenylalanine becoming your tyrosine, then tyrosine becoming your your levodopa and dopamine, etc. Okay, so your neurotransmitters, no, these are synthesized with the help of your tetrahydrobiopterin. Okay, and this is your nitric oxide synthase. Okay, this is your nitric oxide synthase. We synthesize your nitric oxide from your arginine. Okay, from your arginine, and look, now you have your oxygen. And what happens to oxygen? One of the oxygen atoms is incorporated, no, and to your nitric oxide. So what is needed here is your tetrahydrobiopterin as well. Okay, tetrahydrobiopterin, which has always been to be regenerated no, by your dihydropteridine reductase. Okay, so I think that's that. Huh? So what are the four? No, we're done with the four. We're done with the four redox coenzymes. If you're going to study this no, for your quiz or for your shifting exams, you rest. You rest, or you study other things. Okay, you keep. This is for testmanship. Okay, believe me, I've been there. I've done that, been there, okay? We have 11 coenzymes. What you do is you do something else. Okay, you put a temporal boundary between the different groups. Otherwise, it will really be confusing. Okay, so let's now move on. So when you're studying, you stop, and maybe you go back, you absorb. Then eventually, if you several hours later or the following day, what you do is you study the other groups. I study the other groups. So your activation transfer coenzymes, so this is just an example, they usually participate directly in the catalysis by forming a covalent bond with a portion of the substrate. No? So they are bound as well. No? They're prosthetic groups. Okay, for example, this is biotin. Huh? This is the metabolism of biotin. This is your biotin carboxyl carrier protein. So your biocitin, the coenzyme biocitin is what? No, is bonded. Okay, it's tightly bonded to the enzyme. Okay, to the enzyme via certain proteins, etc. Then the tightly held substrate moiety, in this case your carbon dioxide, okay, it will be activated. Kaya nga their activation transfer coenzymes, they don't only transfer the coenzymes but they activate them as well, either for transfer, addition of water, or some other reaction. Okay, so the coenzyme is usually bonded, not to the substrate. Okay. So let's go to the first. So this is the fifth, right? This is the fifth, but this is the first member of your activation transfer coenzyme. So your thiamine pyrophosphate, no, from the term itself, no, it is from your thiamine. I don't have, sometimes it's spelled with the letter E, sometimes without the letter E, no, but let's just spell it with the letter E. So that's your thiamine, that's your vitamin B1. Your vitamin B1, okay? So we don't really have to go into this, no. But your thiamine pyrophosphate, your TPP, is derived from your thiamine, no, via certain, okay, via certain reactions. Our body cannot produce your thiamine pyrophosphate without the vitamins, no, that are exogenously sourced from food, no, mostly from the diet. Okay, most of them are found in the diet. Okay, you have to undergo severe malnutrition in order to be deficient in any of the B vitamins. Okay, so they're found in food, no? Cereals, grains, legumes, nuts, organ meats, beef, pork. No, so vitamin B1 deficiency is rare, no, if there's no malnutrition, because you can find them in many food. However, if in severe malnutrition during war, etc., during famine, what happens? In severe malnutrition, some of the people can have beriberi. Okay, so beriberi sounds like peri-peri. No, beriberi. No, it's a peripheral nervous system disease. No, that's the deficiency. Okay. So let's now go into the chemistry. No, it's a pyrimidine. Okay, it's a substituted pyrimidine. No, anyway, you'll go into your purine and your pyrimidine chemistries. No, you will recognize this is a single ring. Okay, so this is a pyrimidine, part of your nucleotides. No, but this is a substituted pyrimidine ring that is linked to your thiazole. Thiazole. Okay, and the thiazole ring contains your active site. So where is my ano yan? Okay, so for you to remember, maybe the name came from this, no? So your thiamine, the important structure there is your thiazole ring, and they are bridged together. Okay, they're linked together. Okay, by a methylene group, no, methylene group. So this is your thiazole ring. So please do remember the important structural special thing. Okay, what is noteworthy of the structure there? Not what's noteworthy of the structure. For thiamine pyrophosphate, no, it is your thiamine. It's your thiazole ring. It's your thiazole ring. And look, no, it is pyrophosphate. No, pyrophosphate. It's not only phosphate. It's a diphosphate or pyrophosphate. No, two phosphate moieties. This will be the one that is bound to the enzyme. So this is another example of a prosthetic group. No, it's bound to the enzyme, binds tightly to the enzyme. Okay, and this is in order to stabilize this, you need your magnesium ions. Okay, so because of the phosphate groups, we know that are negatively charged. No, in order to stabilize this, you need your magnesium ions. Okay, for its activity. So there, it's in red. Okay, so magnesium ions for its activity, and thiazole ring, thiamine. Okay, so the active site is the carbon 2 of the thiazole. Okay, carbon 2 of the thiazole ring. this is acidic, no, because you have a nitrogen here, no, that is partially positive. So what happens is this is very, very electronegative. The nitrogen is highly electronegative. The electrons are more attracted to nitrogen. So what happens? The hydrogen is easier to give, no? It's dissociable. Once you dissociate your hydrogen, what happens? This become a negatively charged. No? This becomes a this forms your carbon ion, no, carbon ion, negatively charged. This is your carbon ion center. It's electron rich. Okay, it's electron rich. So if it is an electron rich, it can now form a covalent bond with the substrate keto group here. Okay, so this is your reactive atom. It dissociates, right? It dissociates. What happens is this is your carbon ion. No, this is your carbon and ion. So this is highly what? Highly electron rich, no? Electron rich. Your keto group that is attached to your alpha carbon, okay? What happens now? No, it can form a covalent bond, okay? Form a covalent bond, right? With your carbon here, okay, with your alpha carbon, no, alpha carbon is always the carbon that is adjacent to your carboxyl carbon. So what happens, this will now form a covalent bond, and your carbon dioxide will be what will be released, will be released. Okay, so please always remember the substrate keto group, no, covalent bond with the substrate keto group while cleaving the adjacent CC bond. Okay, so this will be cleaved and your carbon dioxide will be released. So the function is it activates, no, it activates and transfers your aldehyde and ketone groups, your aldehyde and ketone groups via carbon ion center and transfers them. And this is just the product that will be stabilized by different resonance forms. Okay, so your thiamine, I know this is a busy slide, huh? This is a busy slide. So just please do remember your thiamine pyrophosphate is what? Do you see this? No, your carbon dioxide is released, no? So it's always, no? It's almost always involved in decarboxylation reactions, okay? Decarboxylation, in non-oxidative decarboxylation and in oxidative decarboxylation. So later on when we go to your PDH, no, your pyruvate dehydrogenase complex, now you will see that you have a pyruvate decarboxylase there. Okay. So you have non-oxidative decarboxylation and you have the oxidative decarboxylation, but it involves your pyruvate as well. So what will be involved here is your TPP, no, thiamine pyrophosphate. So please do remember, cleavage of a carbon-carbon bond next to a keto group. Carbon-carbon bond, okay, carbon-carbon bond that is next to a ketone group, next to a ketone group, right? So this will be cleaved, you release your carbon dioxide. Okay, this will be cleaved as well, no, but this is a more toxic, a more complicated step. Now, your oxidative decarboxylation, this is in your PDH complex. Now, this will release your carbon dioxide and produce your acetyl coenzyme A, which you'll see later on. So, please do remember, no, your alpha ketoglutarate, pyruvate, branch chain alpha keto acid dehydrogenase complexes, no, TPPs are the important coenzyme. Okay, in your pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, your branch chain amino acid dehydrogenase, okay, your thiamine pyrophosphate is the guy, no, is the main guy, okay, is your coenzyme. And there's one more, okay, so please do good thing you have to remember the keto, okay, your thiamine pyrophosphate, keto, okay, because your ketolase, no, your coenzyme for transketolase in your hexose monophosphate shunt, no, or your phosphate pathway, your pentose phosphate pathway, you have a transketolase, it is your thiamine pyrophosphate as well. Okay, anyway, just in case, no, for you, I don't know how to make it easier. No, I know this is really a very very challenging topic, right? TikTok na lang, TikTok na lang tayo, no? So keto, it's thiamine, okay? Transketolase, alpha ketoglutarate, your alpha, your beta chain alpha keto, okay? Acid, okay? Branch chain amino acid alpha keto acid. Ganyan. Dehydrogenases. your pyruvate dehydrogenase, your pyruvate dehydrogenase, etc. It is your thiamine pyrophosphate. This becomes later on more complicated kasi parang it's the same as pyridoxal phosphate. No, a lot of students will really misinterpret or papagbaliktad, no? They mistake one for the other, okay? So thiamine pyrophosphate. So that's that. Next is here, no? Your thiamine pyrophosphate is inactivated by your thiaminase. Thiaminase is an enzyme found in your raw fish, okay, no? Like tuna. Okay, I know it's corny. Okay. Tawa naman kayo dyan sa shoutbox. I feel so lonely. Okay, anyway. Your tuna, your tuna, your thiaminase. So if there's a question, which of the following coenzyme is inactivated by raw fish? Tuna, thiaminase. No? Thiaminase found in raw fish. Okay, so that is your. How are we? Okay, we're here naman, canned tuna, okay. Hindi, pero raw fish, no, yung canned tuna usually ano na yun, eh, medyo cooked na yun, no, kasi it is heat labile. Okay, thank you, Angel. Please remember, ha, please do remember, yung ano, raw fish. This is before known as annulinase, no, I don't know what's the etymological definition, annulinase. Okay, so thiaminase breaks down your thiamine, okay, thiamine, not the thiamine pyrophosphate, but the vitamin itself, okay, into two molecular parts. Next is your pyridoxal phosphate, or PLP. Your PLP. But I'm sure you're familiar with your PLP, no? When you go to your amino acid metabolism, if there's a question about a certain coenzyme, if you don't know the answer, you answer pyridoxal phosphate. This is the most common coenzyme found in your amino acid metabolism, as we will see later on. So pyridoxal phosphate, which vitamin is this coming from? Pyridoxal phosphate comes from your vitamin B6, that is your pyridoxine, okay? Pyridoxine. That's your pyridoxine, okay? Kung paulit-ulit ako, ganun talaga ako mag-lecture, no? I always keep on repeating, always use the word okay, no? Just get used to it, no? So, and I apologize. Usually I repeat, so for emphasis. So vitamin B6 is pyridoxine. And please, guys, we should not exchange the vitamin for the coenzyme. For example, biocytin and biotin. If I'm going to ask you, which is the coenzyme, biocytin or biotin? Which is the coenzyme, pyridoxal phosphate or pyridoxine? Please do remember. Usually it's the more complicated form. Okay, that is the coenzyme. So biocytin is the coenzyme, biotin is the vitamin. Pyridoxal phosphate is the coenzyme, pyridoxine is the vitamin. Okay? So this is synthesized from your pyridoxal phosphate, no, by certain enzymes and not anymore metabolized in the liver. Okay, so if you have INH, di ba, you have in the Philippines, no, we are an endemic country for pulmonary tuberculosis, no, and the primary drug of choice is what, no, is your INH, no, isoniazid. So it is hepatotoxic, no, okay. So sometimes your PLP is rapidly, no, acetylated. It's what? It's broken down, no. So if you have INH toxicity, no, what happens is your PLP becomes deficient. Kaya some of you guys, though, you might be, if you graduated from pharmacy, you might be wondering, in your INH, no, it's always what? It comes always with your pyridoxine, right? With vitamin B6. INH with vitamin B6. It's okay, Mec. There's a recording naman, eh. No, partially you can join the team, team replay. It's okay. It's okay, Mac. So that's that. Okay. So in INH toxicity, so it's here, So INH toxicity, okay, which will lead to peripheral neuropathy, etc. No, so please please do remember. So this is your pyridoxal phosphate from vitamin B6. So the chemistry is, it comes from your pyridoxine, right? It comes from your pyridoxine. Then you just add, no, of a kinase. Now you just add a phosphate group. Okay, so it becomes a pyridoxal phosphate. But sometimes it can also come in the form of pyridoxamine phosphate. Okay, pyridoxamine phosphate. So it can be in two forms, no? Your pyridoxal phosphate and your pyridoxamine phosphate. So it exists in two forms. What is the active site? The active site is the aldehyde group at C4. Aldehyde group, okay? Aldehyde group at C4 of the pyridine ring. Aldehyde group of the C4. This is an aldehyde, right? It's an aldehyde. That is a reactive, no? That's a reactive group. You know very well that an aldehyde and a primary amine, okay, the alpha amino group of an amino acid or substrates, no, or enzymes or what that has an amino acid. For example, most of the time it's lysine, no, because the amino group is in the epsilon, attached to your epsilon carbon. It's far away, right? So usually, no, your aldehyde can interact, no, can react with an alpha amino group and form your Schiff's base, no, Schiff's base. So this is the Schiff's base intermediate, okay, formed by molecular arrangement that determines the type of reaction produced, okay, that is facilitated by your pyridoxal phosphate. So please do remember it is the C for your pyridine ring, because it's the aldehyde group. Okay, it's the aldehyde group. So this is a pyridoxal phosphate or Schiff's base formation. So this is your what? No, this is your aldehyde, okay, and this is the amino group, for example, of your enzyme. No, most of the time this is lysine. So what happens? So you form your imine linkage, no, your aldimine. You form your aldimine. That's your Schiff's base, okay. You form your aldimine. So I really simplify this. Remove all the fluff. Okay, so so this is the shift base. Okay, so what happens here is if this is the substrate, your pyridoxal phosphate will facilitate what? No, transamination. This is for amino acids. This is for amino acids. So your pyridoxal phosphate is really versatile, no, as far as amino acids are concerned, because in most transamination reactions, it is the one. Okay, so it depends, no, on the molecular arrangement, as mentioned here. As mentioned here, no, the Schiff's-based intermediate formed by the molecular arrangement determines the type of reaction that is produced. So in this case, most transamination. By the way, can you tell me which amino acids don't undergo transamination? Doc, we are not yet there. Okay, anyway, no, hopefully, no, when you go to amino acid metabolism, you will see not all amino acids undergo transamination. I think you have two or three that don't undergo transamination. So most transamination reactions, okay, this bond, decarboxylation, ayun na naman, no? Kaya nga this is really confusing because you have certain coenzymes that will facilitate the same reaction, but different substrates. But please, just remember, no, to make things easier, if there's a question regarding a reaction dealing with an amino acid and you don't know the answer, no, just throwing a dart into the into the wall. You pick pyridoxal phosphate, but I'm not telling you it's always the answer, but most of the time, no, because pyridoxal phosphate do facilitate most of the reactions concerning your amino acids. No, their carboxylation, deamination, and racemization. No, racemization, conversion, no, conversion. We'll see later on of your amino acid, no, from L to D, etc. Okay, so if you just remember Schiff's base, no, remember the lysine residue, formation of your imine, no, your aldimine, etc. This is your pyridoxal phosphate. So the function of your pyridoxal phosphate is for transamination, decarboxylation of amino acids, and racemization of amino acids. Okay? Here pala, no? So all amino acids except lysine and threonine, they undergo transamination reaction. So what is a transamination reaction? No? They come in pairs, okay? You know the you know the you know the representative formula of an amino acid, right? You have a centrally located alpha carbon, no? Centrally located alpha carbon, that is your chiral carbon. Then all amino acids will have this, no? It has a carboxyl group that is attached to your alpha carbon, that is your alpha carboxyl group. Then you have your amino group that is attached to your alpha carbon, that is your alpha amino group, and you have your hydrogen, right? Then you have your R group, right? So what happens is you have a corresponding alpha keto acid. Okay, you have a corresponding alpha keto acid. Ketone, no? You have a ketone group. What happens is the amino group will just be transferred to the alpha keto acid and will produce its corresponding amino acid. On the other hand, the amino acid that lost its amino group will be converted to its corresponding alpha keto acid. So you have a group, no? And this is the tandem. This is the tandem that usually accompanies your transamination reaction or your alpha ketoglutarate glutamate. Okay, they're the most common. Okay, it's one of the alpha keto acid amino acid pairs. So your glutamate is a source of your amino group for your transamination reaction, and the recipient will be your alpha ketoglutarate. Okay, etc. So in one way or another. So your pyridoxal phosphate is also involved in racemization of your amino, no, or racemization of your amino acids from your L to D. No, these are your enantiomer. You know very well that your amino acids are mostly in the L, diba? In the L enantiomeric form. So what will racemize or that will convert from L to D will be facilitated by your pyridoxal phosphate as well. Then the carboxylation of amino acids is a long process, no, but your decarboxylation is also what facilitated by your pyridoxal phosphate. Okay, I know, no, it's doc, it's many. So my advice really is don't study the 11 coenzymes in one sitting. No, you will lose your mind. You will lose your mind. You study them according to groups, no? According to groups kasi they're related. So the reactions catalyze is your aminotransferases, no? Exemplified your ALT, AST, your amino acid decarboxylase, amino acid racemase. Please do remember if it is regarding amino acids, a very important coenzyme that will participate will be your pyridoxal phosphate. Okay, the next is your coenzyme A. Coenzyme A, which vitamin? Which vitamin is your coenzyme A derived from? Which vitamin is your coenzyme A derived from? Doc, is it a vitamin derived coenzyme? Yes, no, yes. Ang galing, no, Thea. Very good. No, but I'll tell you a very easy way to remember, okay? Sige. So it's from vitamin B5, no? Pantothenate. Pantothenate. Kasi for me, when I was a student, I didn't know this, no? For some, this will be the first time that they will hear. Kasi ang uso talaga, what's really familiar is niacin, riboflavin, folic acid, etc. No, but you have vitamin B5, you have vitamin B7, you have vitamin B9. Okay, so vitamin B5 found also in a lot of your food, but it's an essential vitamin. So it's synthesized in a 5-step process, no, that requires ATP, not necessarily that. No, but how do you remember? So coenzyme A is known as CoA or CoASH. Okay, ni sulfur here, CoASH. No, so if you're going to count the number 1, 2, 3, 4, 5. So that's vitamin B5 and it's 5, no, parang pentatonics, no. I love that group, no, pentatonics. So it's your pentathenic acid, no. So it's your pantothenic acid, but more or less that can help you remember. Okay. I know it's corny, I know it's corny. I know it's stretched, but if it can help you, why not? No, if this can help a student, then I'll really be very happy. Okay, so pantothenic acid is vitamin B5 is the source for your coenzyme A. Para when it goes down to the wire, no, ano nga ba yun? Ano nga ba yun? Then hindi ka na mapakali, right? So, alam ko, CoASH, five-letter word. Vitamin B5 pentathenic. So, it's pantothenic acid. Okay, so I hope, so, you can use other memory aids if you want. Okay. So this is a nucleotide as well, no, composed of your adenosine 3 monophosphate, Okay, then you have your pyrophosphate, you have your pantothenic acid, and your thio. No, if you see thio, that means it's a sulfur. No, ethanolamine. Okay, so please do remember the structure. Doc, we have a lot of structures. I know, but please do remember it's already here. Now I put this on the left side. You just studied this. I hope this will help you. Now I summarized everything. I summarized everything so it will be easier. So you have the different components, but what's important here is the sulfur as well, okay? Because the active site is the sulfur, no, in your thioethanolamine group, you know very well that sulfur is a highly electronegative atom, no? Electronegative means it has the greatest attraction to the pair of electrons, okay? Greatest attraction to the pair of electrons. So it's what, no? So it is a strong nucleophile. Okay, it's a strong nucleophile. In other words, it will look for regions that are devoid of electrons, right? That are devoid of electrons. So it will now attack your carbonyl groups and form acyl thioesters. So meron ba ako reminder dito? Okay. So this is actually the etymological definition or the origin of the term coenzyme A. The A stands for acyl or sometimes activation of your acetate. No, activation of your acetate. Okay, so it's coenzyme A. So if you see a coenzyme A, I know it's your acyl groups, no, acyl groups, acetyl, succinyl, or fatty acyl to form thioesters, no, because you have your sulfur. So if we're going to look at this, no, these are the more or less the functions of your coenzyme A. It is involved in your fatty acid synthesis and oxidation. No, it's involved in what conversion of your pyruvate, no, to your acetyl coenzyme A. So it's you can find that in your pyruvate dehydrogenase complex as well, and it is involved in your lipid metabolism, no, because you know your acyl groups, no, fatty acyl groups, they're involved in lipid. Okay, so lipid metabolism, fatty acid synthesis. Okay, so that's that. So as I've mentioned, the A in CoA stands for acyl group that becomes attached or activation of your acetate. So here from here we can see very well. Okay, so acetyl coenzyme A synthetase, pyruvate dehydrogenase complex. Now you see that your coenzyme A is involved in this. So your fatty acyl coenzyme A synthase, coenzyme A synthase or synthetase. I think it's synthetase. No, my mistake. Synthetase because you have your ATP. No, in terminology, no, I know it's not always right. Now, if you see what's the difference between synthase and synthetase, if it involves energy, no, ATP, it's synthetase. So your fatty acyl coenzyme A synthetase, okay, it's facilitated by your coenzyme A, and coenzyme A, you find that as well in your PDH complex, no, in dihydropoyl transacylase. You'll see that. Okay, so it's involved in your lipid metabolism. So that's your coenzyme A. Biocytin. Biocytin. I think this is the last, no, of your activation transfer coenzymes. Now we're progressing very well. Biocytin, is this from a vitamin? From which vitamin is biocytin? From which vitamin? Thea, from which vitamin is biocytin from? Not a lot of people will know this or will remember, okay? But it is from vitamin B7. Very good, no? Vitamin B7, biotin, no? Biotin is vitamin B7. Please do remember, biotin is the vitamin, biocytin is the coenzyme because the coenzyme is always more complicated than the vitamin. You add on to the vitamin. You add on to the vitamin. Biotin is your vitamin. And your biocytin is the coenzyme. Siguro for you to remember is biocytin 7. Okay. Biocytin, this helps me remember, no? Biocytin 7 is vitamin B7. Okay, biocytin, I know it's corny, but that's really up to you. You may not use this. Vitamin B7. Okay. Can I have a shout out? Corny ba? Or nangingilipit na ba kayo sa cornyhan? Anyway, but I hope no, that this will help you remember. So you have your biotin vitamin, okay? Biotin vitamin. No? So you have your biotin plus you have your lysine. Again, no? From here you can see that in your coenzyme chemistry, your lysine really plays a very important role, right? The amino group, no? The epsilon amino group. You have a amide, okay? You have an amide group, no? That joins, no, your biotin to your lysine, and this is the enzyme. Okay, this is the enzyme part. So this is tightly bound as well, huh? So this is tightly bound as well. Okay, so the synthesis, no, we didn't go into the synthesis, but please do remember, no, sometimes nga, if for example, I think, no, in some bodybuilders, no, they keep for the albumin, no, they take in a lot of raw egg whites, right? And this will inhibit what? The formation of your biocytin from your biotin. Okay, so your avidin, take note, ha, it's here. So yung others, these are just additional information. Excuse me. Okay, so your avidin will inhibit the formation of your biocytin from your biotin. For the for the chemistry, for the chemistry, it is the imidazole. No, this is your imidazole. No, it's an imidazole ring fused with your tetrahydrothiophene. This is your tetrahydrothiophene linked to your valeric acid. Doc, you have a lot of, yeah, I know, no, you have a lot of components. So please study them. You cannot survive on this by reading this once, okay, or watching this video once, okay. So you just take particular note of the peculiarities or the important features, no, of each structure. Okay, if you see imidazolone, if you see your tetrahydrophene, you have a sulfur, and your valeric acid, that is your biocytin. And again, it is bound not to the apoenzyme by the epsilon amino group of your lysine. So that is the chemistry. The active site here is what? No, the active site here is the nitrogen. No, it's the nitrogen. The sulfur atom will just polarize the urea nitrogen. No, this is the urea. The urea. No, this looks like the urea. No, but this will now what? No, polarize this. Okay. So what happens here is your carbon dioxide will now bind, no, to this nitrogen. So the sulfur atom of your tetrahydrophene tetrahyd oh no, it's yeah, the sulfur atom in your tetrahydrothiophene, okay, will polarize this nitrogen and will susceptible to attack by a carbonyl group. Okay, so this what will be transferred here is your carboxyl group to nitrogen. So you saw this kanina, right? So the function here is activates and transfers carbon dioxide. So from here, now you have your bicarbonate. You have your bicarbonate. It will now be attached to the urea nitrogen, right? To the urea nitrogen of your imidazolone group, to form your carboxybiotin, right? Your carboxybiotin. Then this carboxybiotin, your carbon dioxide now will be transferred to your acetyl CoA to make it one carbon longer. It will now become your malonyl coenzyme A. Acetyl coenzyme A to malonyl coenzyme A. So on tip here, biocytin is involved in carboxylation. In carboxylation. We saw two coenzymes before that is involved in decarboxylation, right? I hope you remember your thiamine and your pyridoxal phosphate. No, they're decarboxylation. But your biocytin is carboxylation. So biocytin, baliktarin mo, it's carbon dioxide. No, you add your carbon dioxide, no, biocytin. Kasi believe me guys, no, when you have the 11 coenzymes, all in all, halo-halo, no, in 8 questions or 10 questions, what is biocytin for? Is it carboxylation, decarboxylation? Just remember this. So biocytin, it's adding carbon dioxide. Adding carbon dioxide via your N-carboxybiotin. When in doubt, pyridoxal phosphate. But not all, right? In amino acids, Thea, in amino acids. But for carboxylation, especially, no, extending your fatty acyl, okay, becoming malonyl coenzyme. This is in your fatty acid synthesis. Okay, so you need your biotin. Carboxylation. Okay, so hopefully, so synthesis of malonyl coenzyme A from acetyl coenzyme A, pyruvate carboxylase, no, pyruvate carboxylase, propionyl carboxylase, methyl malonyl coenzyme A, carboxylase. So these are all carboxylation. So carboxylation, please do remember this yellow thing here. Okay, that's how I remember it as well. Okay, so when I go into yung small group discussion before. Okay, so by citing automatic, no, carboxylation. Okay, so last three, last three. Then we have several questions. Okay, I apologize. No, this is taking longer, but I cannot make this faster. I cannot make this faster because there's so many. Dok, buti nga 11 lang eh. Buti hindi amino acids 20, right? Right? No, count our blessings. Okay. Anyway. O transfer coenzyme. So basically, what they do is they just transfer. Okay, they just transfer. So you have three, no? The last three. Lipoic acid, tetrahydrofolic acid, cobamide coenzyme. Which among the three is not derived from vitamin? Which among the three here is not derived from vitamins? Baka sasabihin mo, Dok, yung tetrahydrofolic. No, no. Ano yun? Tetrahydrobiopterin yung T, no? Isipin mo yung TLQ, true lovers quarrel, no? Two lovers quarrels. Tetrahydrofolic acid, no, is from folic acid, is from folate, right? Which is a vitamin B. Cobamide coenzyme, we know is from vitamin B12. These are lipoic, no? That is the L. That is the lover. So let's start with your lipoic acid. The better term is lipoamide, lipoamide, because it is your lipoic acid, free lipoic acid does not exist. It's always an amide, no, with a certain amino acid. Okay, so it is not sourced from any vitamin. It is sourced from your octanoic acid. It is sourced from octanoic acid or your caprylic acid. No, the classical name is caprylic acid. Okay, but the scientific name is your octanoic acid, an eight carbon saturated, no, fatty acid. Okay, then you have certain steps. You have certain steps. Now it is synthesized via fatty acid biosynthesis. Okay, then you now form your, then you exchange, no, you exchange your hydrogen with sulfur. Okay, so you now form your lipoamide. No lipoic acid is produced, no, so it's always what, no, it is always attached as an amide, no, to the lipoyl domain protein. So lipoyl synthase. So just remember, it is synthesized from your octanoic acid via fatty acid biosynthesis. Or if you see caprylic acid, lipoic acid. Okay. Anyway, siguro acid, octanoic acid, caprylic acid. No, hopefully that will point you to the right direction. Okay, when you're answering. Okay, testmanship. So, but the better term is lipoamide. So the chemistry, ayan, TLQ, no? So true lovers quarrel, tetrahydrobiopterin, lipoamide, lipoic acid, and your ubiquinone are not vitamin source, are not sourced from vitamins. This is the last, okay, of the three. So it's an alpha lipoic acid, no? It's an organosulfur compound. No, you have your sulfur groups, no? The scientific name is your 6,8-dithiooctanoic acid. No, it comes from your octanoic acid. Dithio, no? You have two sulfur. You have two sulfur. It's also known in shorter terms as your thioctic acid. Thioctic acid, and it is bound again, no? It's bound, it's bound to the enzyme via, no, via an amide bond, no, to the epsilon amino group of your lysine, so that's why it's known as your lipoamide. So it's a prosthetic group, covalently bound to the lysine residue of the enzyme. So the active site is the disulfide group, no, yung sulfhydryl group. So it can be in the oxidized form, and it can be in the reduced form. If it is your sulfhydryl, that is your reduced, no, that is your dithiol, thiol reduced, okay, dithiol, and this is your disulfide, okay, that is your this is your oxidized. So if it is cyclic, no, disulfide, that is your oxidized, but if it is reduced, this is your dithiol. Doc, why is this not included in the redox? Because they just don't transfer electrons, they also transfer acyl groups as well. Okay, they transfer acyl groups as well. So it was included maybe in the transfer group, not just solely in the redox, no, in the redox coenzyme group, the first group, because it accepts and transfers acyl groups as well during oxidative decarboxylation of alpha keto acids. Ito na naman, no? Parang this looks like similar. No, this looks similar to what we have studied, no? But this is what, no? This is um transfers your acyl group plus electrons. Transfers acyl groups plus electrons. Oxidative decarboxylation of alpha keto acids. No, you can see here. Okay, it's found in your pyruvate dehydrogenase complex, alpha keto glutarate dehydrogenase, your branch chain alpha keto acid dehydrogenase as well. Okay, so this, no, from here you can see. Okay, you need your TPP, no, but you need your lipoamide as well, no, to transfer the acyl groups. So this is, I think, no, I've read it in one book. No, this is unique. This is unique because we know we have learned that your coenzymes have various, no, have variable specificity. No, they're not really specific to a substrate. No, but your lipoamide, I think, is specific to your alpha keto acid dehydrogenase complexes. Okay, so you know your for example your pyruvate dehydrogenase complex, your branch chain amino acid dehydrogenase complex, right? And your alpha ketoglutarate dehydrogenase complex. Now this involves oxidative decarboxylation. Oxidative decarboxylation. So this is exemplified by your pyruvate dehydrogenase complex. You will study this in your carbohydrate metabolism. Now, your PDH, your pyruvate dehydrogenase complex, bridges your glycolysis and your tricarboxylic acid cycle. It converts your pyruvate to your acetyl coenzyme A via oxidative decarboxylation. It has basically, I think, 3 enzymes and 5 coenzymes, right? But we're getting ahead of ourselves, no? But in your pyruvate dehydrogenase complex, now you have your E1, E2, you have 3 enzyme groups, okay? But you have 5 coenzymes. Now the 5 coenzymes are, you have your what? No, your thiamine pyrophosphate, you have your lipoamide, you have your coenzyme A, you have your FAD, and you have your NAD. Now you'll study this in your carbohydrate metabolism, okay? But in your PDH, this is responsible, no, for your oxidative decarboxylation as well. I know it's a little confusing, So your lipoamide specific to your alpha keto acid dehydrogenase transfer reactions. So alpha keto acid dehydrogenase transfer reactions. So pyruvate dehydrogenase complex, alpha keto glutarate dehydrogenase, your branch chain alpha keto acid dehydrogenase. I'm not familiar with this much. Okay, so that is your lipoamide. So it's summarized here in this table. Now what you can do is siguro you print this, you cut it out, etc. If you find no disclaimer, no, this might not be totally accurate. Maybe I've inputted some information, but I checked them. But if you find something that's wrong, you let me know. Okay, para I can put a comment no in the video, etc. Okay. Your second to the last, number 11, no, yung tetrahydrofolic acid, your FH4. Sometimes this will be abbreviated as THF, okay? THF. Doc, hindi BTS. No, THF, FH4, FH4, okay? So your tetrahydrofolic acid, no, from the term, it's your vitamin, it's from your folate. It's from your folate, okay? Vitamin B9. Doc, how will I remember vitamin B9, folate, vitamin B9? You count the numbers of letters in folic acid. FOLIC ACID. 9 ba yun? Tama ba? Folic acid, 9 letters. Vitamin B9. Talagang pilit na pilit. On a 7, B7, anyone? What's B7? Vitamin B7. Vitamin B7. Tama. Thank you, Thea. Vitamin B7. Di biotin, bio7, biotin. Okay. Anyway, bahala kayo. Basta if it can help you. Okay, anyway. So your folic acid is composed of 3 parts. Now you have your pteridine ring. Then you have your para-aminobenzoic acid and your glutamate. This is polyglutamate because you have a series of glutamic acid. Series of glutamic acid, polyglutamate tail. Okay, so it's synthesized, no? Your tetrahydrofolic acid is synthesized from your folate no, via reduction. Okay, reduction of your dihydrofolate reduction, DHFR, dihydrofolate reductase. Okay. Sige, no? So, medyo confusing 'to with your BH4, no, yung Tetrahydrobiopterin, kaya nga don't study them at the same time, ha? So, sometimes nalilito rin ako dito yung Tetrahydrobiopterin and Tetrahydrofolic Acid, okay? What is involved in hydroxylation of aromatic amino acids? What is involved in hydroxylation of aromatic amino acid? Hydroxylation, no, yung Tetrahydrobiopterin kasi hydro, pero Doc hindi ba hydrofolic acid? Kaya please take note of the distinction, ha? Don't study them at the same time. Tama, Thea, no, it's Tetrahydrobiopterin. Okay, no, so you form your tetrahydrofolate. So, pyridine ring plus PABA plus glutamic acid. So when you're going to study this, no, for each coenzyme, you have different components. So you always go back. You always go back, or maybe you post it on the refrigerator, you post it on your bathroom, and you look at it always. It's very hard, really. It's very hard to remember, no, the different components. What has a thiazole ring? Thiazole ring that contains the active site. Thiazole ring. Which contains the imidazolone ring. 'Di ba? Pag halo-halo na, if they're bundled together, it's really very confusing. Thiazole ring is thiamine, 'di ba? Meron tayong parang memory aid for that. Okay, so what's the active site of your tetrahydrofolic acid? Please do remember it's N5. N5 of your pyridine ring, N5 of your pyridine ring, and N10. This is wrong, ha? It's N5, the shortcut, no? N5 of your pyridine ring and N10 of your PABA. N10 of your PABA. N5 of your pyridine ring and N10 of your PABA. Okay, so they have highly available lone electron pairs for attachment of one carbon groups. Attachment of one carbon groups. So for in this case, now this is your tetrahydrofolate, right? So it's already reduced, no? Your tetrahydrofolate. What happens is that they take in different forms of your carbon, one carbon group, no, one carbon group. So this is the most oxidized. This is the most oxidized formyl, and this is the most reduced, your methyl. If we're going to look at this, I'll show you the structure later on. So from formyl, it went into methanil, that it went into methylene, then into methyl. So this is what? No, this is reduction. This is reduction until you go into the methyl. Di ba, what did we say? What did we say? If you have the most number of hydrogen, you're the most reduced, right? Because you contain the most number of electrons. Di ba I told you that kanina? And why is this the most oxidized? Why is this the most oxidized? It has the least number of hydrogen and it has an oxygen, right? Remember, addition of oxygen is oxidation. Loss of electrons in the form of hydrogen or addition of oxygen is oxidation. So formyl is the most oxidized, followed by methanol, right? You have still the hydrogen, but you don't have the oxygen. Then if this is going to be reduced, you have your methylene, you have two hydrogen atoms, then you have your methyl. Methyl, you have the three hydrogen atoms. This is the most reduced. In the reduction steps, no, this is irreversible. Okay, it's once you form your methyl, no, your N5 methyl FH4, it cannot go back anymore. So what accumulates in the cell is your N5 methyl FH4. I've mentioned, I've mentioned your carbon will attach to your N5 and N10, right? It will attach your N5 or N10 or to both. Look, in this case, it is attached to both. It's attached to both. Then this is attached only to your N5. Kaya from here, it's a little confusing, right? It's N10 formyl. This is C double bond OH, right? Then it's attached to your N5, to your N10. It's your methanol, C double bond, single bond. That's your methanol, N5, N10. Kaya nga you have two nitrogens, right? Then you have N5, N10 again, methylene, it's CH2. Then boom, now you have the most reduced, that is your methyl. Okay, so please do remember, And these are one carbon groups. One carbon groups. How do you remember the tetrahydrofolic acid will transfer one carbon groups? What I do is I remember the F, formyl, folate, formyl. Formyl I remember. Formyl I remember is a one carbon group. So para mabilisan, right? Para if you're under 60 seconds, if you're under pressure and you don't know anymore what to do, is this tetrahydrobiopterin or is this tetrahydrofolic? Folic, 4 mil, no? So 1 carbon groups, right? 1 carbon groups. So it depends, no? So it depends on what 1 carbon group, and this is what will form your 1 carbon pool. So which form of your FH4 will be involved depends on the type of carbon, okay? The one carbon group that is transferred. So the reactions, I think I've looked into this. You more or less you have three. Okay, you have three, but I'm sure there are more. No, but this is the most common. The reactions, the function is the transfer of one carbon groups. Now we've been here. The reactions catalyze is what? No, the formation of glycine from serine. Look, what is lost? What is lost? No, you lose a methyl group plus a hydro. No, so this is your serine hydroxymethyl transferase. Okay, serine hydroxymethyl transferase. Anyway, methyl is there. No, that will give you a clue. Hydroxymethyl transferase. So you lost your methyl group. Boom. No, that is what facilitated by your tetrahydrofolic acid. And what is this? What is the difference between uracil and thymine? I know you're not yet in your nucleotide chemistry, right? In your purine and pyrimidine chemistry. But the difference between uracil and thymine is that thymine is just a methylated uracil, right? So a methyl group. No, you add a methyl group to your uracil to your deoxyuridyl monophosphate. Deoxythymidine monophosphate. Okay, so you also add a methyl group. No, so in this case, what is involved here is your N5-enten methylene. Okay, so what is the coenzyme that will participate in this is tetrahydrofolic acid because these are one carbon groups, right? Um, So your methotrexate, your sulfonamides, okay, will inhibit your tetrahydrofolic acid in various ways. Okay, either the thymidylate synthase. Now this is your thymidylate synthase. That's letter B, or it will inhibit your dihydrofolate reductase. So you'll not regenerate your tetrahydrofolate. So when you see methotrexate and your sulfonamides, so methotrexate I think is an anti-neoplastic drug, right? It is an anti-metabolite. No, these are anti-folates. These are anti-folates, so anti-metabolites. So same with sulfonamides. This will inhibit your tetrahydrofolic acid. Okay, your formyl transferase. Do I have a slide? Okay, your formyl transferase, although this is a minor one, this is involved in purine metabolism and synthesis. No, purine metabolism and synthesis. Okay, anyway, formyl is there. Okay, formyl, tetrahydrofolic acid. So more or less, now you can make the connection. Last. Okay, cobamide coenzyme. Thank God. Okay. Cobamide coenzyme from which vitamin? Cobamide coenzyme from which vitamin? It's from vitamin B12. Thank you, right? Thank you, Thea and Angel. Okay, so I don't have any memory aid for that. No, but you know this, vitamin B12. I'm sure, no, you know that it is cobalamin. Okay, it is important in your DNA synthesis, right? Um, anyway, I forgot. No, I forgot. In your cancer in anti-neoplastic, no, this is because this is what involved in DNA synthesis. Okay, your thymidylate synthase is involved in DNA synthesis, so this is in really very important in cancer, no, cancer therapy. Okay, this, I think this is a chemotherapeutic agent, so in a way hopefully it will stop the progression of in cancer cells, etc., the spread, etc. Okay, in the same way, your vitamin B12 is important as well in your DNA synthesis. Okay, so yung anemia, no, yung formation of the red blood cells, etc. If there's a question, no, megaloblastic anemia, pernicious anemia is more for vitamin B12 deficiency. But sometimes it is also pointed to your folate, no, tetrahydrofolate. But if you're given the two choices, no, if there's a question about megaloblastic or pernicious anemia, you either look for cobalamin, that's a priority, folic acid if cobalamin is not there, okay? Because your tetrahydrofolate or folate deficiency can also lead to what? Um, Pernicious anemia. One more thing. Folate is given to pregnant women right? Di ba? And folic acid, aside from iron, is given for pregnant women. Why? Because it's important nga in synthesis of tissues, right? So yung mga neural tube defects, right? Neural tube defects. Okay. No, yung mga neural tube defects, yung mga congenital anomalies, etc. No, that's for folate deficiency, folic acid deficiency. Anyways, as I mentioned, na your vitamin B12, your Cobamide coenzyme for megaloblastic anemia because it's very important in maturation of your red blood cells as well. So chemistry, you have it's a tetrapyrrole ring. Now, what does this remind you? If you have a tetrapyrrole ring, it's just like in hemoglobin, but the main difference is in hemoglobin, no, your pyrrole rings, no, your four pyrrole rings are bridged by your methylene bridges. Okay, methylene bridges, methynyl bridges, not C double bond C single bond, methynyl bridges. However, in this case, no, you have two pyrrole rings that are bridged, that are joined directly. So that's the difference, no? And instead of iron, you have cobalt, right? Tama, heme group, no? Angel and Thea, no? Heme group. Just you can find that in heme, right? But instead of iron, you have cobalt. Now you have cobalt. So your cobalt is what? No, it's complex. No, it's complex with six, no, different ligands. No, you have four nitrogen atoms. Then this is very very important. No, you have your dimethyl benzimidazole, dimethylbenzimidazole moiety, and that last, the six is connected to an X. No, the X can be your cobalt can be bonded to a carbon. Now the carbon can be found in methyl. So if the X is methyl, that is your methylcobalamin. If the X is your deoxyadenosine, it is deoxyadenosylcobalamin. No, so that's the source of your terms. Now, why do we have methylcobalamin that is specific for a certain reaction, and you have your deoxyadenosylcobalamin that is specific for next reaction? Is it depends on the substituent for your X. So please do remember the chemistry. Ha, you have a choline ring. Actually, that's a choline ring, no, because of the cobalt. Sa tetrapyrrole, you have your dimethyl. Okay, dimethyl, two methyl groups. Okay, dimethyl benzimidazole, no, benzimidazole. Then you have the two carbon containing moieties. No, it can either be a methyl or a deoxyadenosine that is attached to your cobalt, no, in the six connection. Okay. So the function is dalawa lang, no? I think this is the enzyme that is only involved in two reactions. First is you have this is your methylcobalamin, no, methionine synthase. No, I think Thea was suggesting this, no? Okay. So in order to synthesize, no, your methionine, okay, from or to regenerate, no, your methionine from your homocysteine, no, you need a source of your methyl. No, you need a source of your methyl, and the methyl here will be brought to you by your Cobamide coenzyme from your N5-methyl tetrahydrofolate. No, the most reduced, right? Okay, so this is your methionine synthase or your homocysteine methionine methyl transferase. Okay, transfers a methyl group. And last, you have only two reactions. Remember, your vitamin B12 only facilitates two reactions. No, synthesis of your methionine from your homocysteine, methionine from your homocysteine. Okay, and your methylmalonyl coenzyme A mutase. No, in other words, mutase is what? No, rearranging. No, rearranging your methylmalonyl coenzyme A so that it will become your succinyl coenzyme A. So this is via your adenosylcobalamin. So it's different, adenosylcobalamin. So I think, please do remember, if it is your methionine synthase, it's your methyl. It's very important because it's the methyl group that is being transferred on your methylmalonyl coenzyme A mutase. No, it's your adenosylcobalamin. Okay, so the arrangement of your L-methylmalonyl coenzyme A to form succinyl coenzyme A. And you will see this later on in certain processes. Okay, in your branch chain amino acid. Okay, branch chain amino acid metabolism, no, you will see this. And in your add chain fatty acids, no, add chain fatty acids, most fatty acids in the body is even numbered. However, your add chain fatty acid, the last, okay, will be propionyl coenzyme A. Okay, not acetyl coenzyme, but propionyl. They have to be converted to your succinyl coenzyme A via this. Okay, so we have 11. Huh? You have 11 coenzymes. You have 4 from your redox coenzymes. You have 4 from your activation transfer coenzymes, and you have 3, no, for your transfer coenzymes. Are we ready for some sample questions? We have 32 na lang, no? I do understand, no? We're almost 2 hours. But hopefully, no, from what we learned, hopefully we can answer some. Huh? But if not, no, we can go back or look into our notes and then look why. That was the answer. Okay, so question number 1, which of the following statements, ready, not tayo ready, which of the following statements correctly describes reduction of one of the electron carriers, NAD or FAD? Letter A, NAD accepts 2 electrons as hydrogen atoms to form NAD2H. Letter B, NAD accepts 2 electrons that are each donated from a separate atom of the substrate. Letter C, NAD accepts 2 electrons as a hydride ion to form NADH. And letter D, FAD releases a proton as it accepts 2 electrons. Siguro, may nawala na putol. What's the answer? What's your answer? Hello. Right, Angel. No, right. It's right. Okay. NAD accepts only one hydride atom, hydride ion, but it contains 2 electrons. Where did the other hydrogen go? So it goes into the solution. Kaya nga NADH plus H, right? NADH plus H. Okay. So let's review. No, your nicotinamide adenine dinucleotide, your NAD, your NADP will transfer electrons in the form of hydride ions. Okay, it's a hydrogen atom plus 2 electrons. Your FAD will accept 2 electrons in the form of 2 hydrogen atoms. Kaya nga siya FADH2, FMNH2. Your ubiquinone will transfer electrons in the form of hydrogen atoms. Kaya nga, di ba, quinol. Okay, QH2. So it's only NADP that is different. Okay, 2 electrons in the form of your hydride ion. So it's letter C. Question number 2. Which of the following coenzymes facilitate the transamination, decarboxylation, and racemization of amino acids? Go. parang kahoots, no? Go. Anyway, we're dealing with amino acids. So what's your answer? Ubiquinone has nothing to do with ubiquinone has nothing to do with metabolism of your amino acids, right? Because ubiquinone is a redox coenzyme. It participates in the transfer of electrons. Tetrahydrofolate, what is tetrahydrofolate for? For formyl, no? This is a transfer of 1 carbon groups. Cobamide coenzyme, we know that this is only involved in two reactions, the methionine synthase and your methylmalonyl coenzyme A mutase, right? So it's pyridoxal phosphate, right? Remember this, no? This is pyridoxal phosphate, no? Yung imine formation, right? It depends, no? What is rearranged, what is formed to be able to get the reaction, no? Transamination, decarboxylation, racemization, etc. of your amino acids. So it is letter B. When in doubt, pag amino acids sa iha, don't quote me. Baka naman. Sabi ni Doc Motis, for amino acid metabolism, most of the time it's pyridoxal phosphate, but not all the time. There might be some caveats, okay? Huwag niyo ako sisihin eventually. But for amino acids, the most prominent coenzyme is pyridoxal phosphate. Question 3. Which of the following coenzymes is unique to alpha keto acid dehydrogenase complexes? is unique to alpha keto acid dehydrogenase complexes? NAD might be, right? But it is not specific only, right? It's not specific only. Kasi in pyruvate dehydrogenase complex, you have NAD, but you also have NAD in complex 1, right? So hindi pwedeng NAD. Biocytin, what is biocytin ba? Where do you find biocytin? If you remember, biocytin, OCCO is carboxylation, right? So hindi rin siya pwede, di ba? Thiamine pyrophosphate. What is involved in thiamine? Thiamine, you have your thiazole ring. What does thiamine pyrophosphate do? It is involved, right? It is involved in your pyruvate dehydrogenase complex, right? But it is also involved in other reactions, right? Di ba we said something that something is unique to your pyruvate dehydrogenase complex, to your branch chain amino acid alpha keto acid dehydrogenase, to your alpha keto glutarate dehydrogenase complex, and it is lipoic acid. Okay, and it is lipoic acid. Lipoic acid is involved to what? No, is involved in transferring electrons plus your acyl groups as well. Huh? So that is what will that is the delineation. That is the distinction. Okay, lipoic acid does transfer. I think not. It does transfer your electrons, but it also involves your acyl groups as well. Okay, yan lipoic acid. Very good, very good. Ian, ha? Very good. Ian Cedric Tan. Sige. O yan, the active site of FAD is found in the structure of the coenzyme. Ayan, sabi ko sa inyo, yun yung mga components na pampagulo, right? Where do you find thiazole ring? Ano, guys, where do you find thiazole ring? I know, madaling question, di ba? Kasi alam natin yung sagot kagad. Pero if I'm going to ask you, where do you find thiazole ring? Thiazole, thiamine, right? So it's found in thiamine. Where do you find imidazolone? Where do you find imidazolone ring? Saka where do you find tetrahydrobiopterin ring? So please remember the different components. I know the question might be a little giveaway, no? But your imidazole ring and your tetrahydrobiopterin ring, you see that in Biocytin, okay? You see that in Biocytin. Isoaloxacin ring, yes, no? This is a Flavin. These are Flavin nucleotides, no? Yung three rings, okay? Then you have your active sites here that can accept or give away an electron. Then you have a quinone and you have a quinol, right? Diba? These might be sources of your reactive oxygen species, but no, in complex 2, they're tightly bound, right? They're tightly bound to protect. Ang source talaga ng reactive oxygen species will be the ubiquinol, no? Because the same ano yan, same principle. But your ubiquinol is mobile, no? It's permeable in the lipid member, no? Electrons can easily escape. Very good, no? Very good. Almost got it, no? Shout out to Timothy, Matthew Hernandez, to Jesly Anton. Okay. Tama, no? Timothy, Matthew Hernandez. No? Your imidazolone is your biocytin, right? Question 5. In the oxidative decarboxylation of pyruvate acid to acetyl-CoA. So you know what is that process? Oxidative decarboxylation of pyruvic acid. Oxidative decarboxylation of pyruvate to acetyl-CoA. What is that process? Or what is that complex? Oxidative decarboxylation of pyruvate to acetyl-CoA. Pyruvate is the product of glycolysis, right? It has to be converted to something so that this moiety can enter your TCA cycle. No, it has to undergo oxidative decarboxylation via your pyruvate dehydrogenase complex. Very good, no? Ayan, Cedric, no? Pyruvate dehydrogenase complex. So ang tanong, the question is, which of the following is not involved in your pyruvate dehydrogenase complex? Thiamine pyrophosphate, Pyridoxal phosphate, FAD, lipoic acid. From what we learned kanina, you know very well, di ba? What pyridoxal phosphate is mostly involved in which? Pyridoxal phosphate is mostly involved in yan nga, di ba? This guys, this is what I'm telling you. Because pareho silang phosphate, di ba? So mentally parang parang it's hard, no? You can confuse one for the other, right? Di ba? Magkasunod pa sila. And they also belong to the same group, right? if you're going to look at your pyruvate dehydrogenase complex, what do we see? This is pyruvate, no? This is your saan na ba acetyl? Ayun yung acetyl coenzyme A, no? That's your acetyl coenzyme A, right? It has three enzymes, no? Your pyruvate dehydrogenase, your dihydrolipoyl transacetylase, and your dihydrolipoyl dehydrogenase, E1, E2, E3. You will study this in carbohydrate metabolism. Don't worry. But we can see here you have 5 coenzymes. No, you have your thiamine pyrophosphate. You have your ano to? Ano to? Yung merong may thioctic. Yung merong dithiol. What's this? Yung merong sulfide, disulfide, and sulfhydryl. May dithiol. This is your lipoamide, right? That's your lipoamide. Then you have your coenzyme A, kaya sa acetyl coenzyme A. Then you have your FAD, and you have your NAD. So what is not belonging here? What's not belonging here is your pyridoxal phosphate, because we know that pyridoxal phosphate, no, formation of your Schiff's base, etc., is what? No? Yung, is the reaction of your acetaldehyde with the amino group, no, of an amino acid that is in the substrate. You form imine. Depending on the rearrangement, it can either cause transamination of amino acids, decarboxylation of amino acids, deamination, substitution, elimination, racemization, right? So pag pyridoxal si Tay. Sandali, pakita nga natin yan. Ay, sorry. Ayan, when in doubt nga pala, it's pyridoxal phosphate, not all the time. Okay. Anyway. So, but please, siguro, make it a point na lang, guys. No? Please remember the 5 coenzymes that you find in your pyruvate dehydrogenase complex as early as now. Okay, so you have thiamine pyrophosphate, you have your coenzyme A, you have your lipoamide, you have your FAD, and you have your NAD. Okay? Okay, question number 6. Question number 6. Ayan, an individual with INH toxicity will most likely have an impairment of which reaction? Ay, alam ko yun, Doc. That's pyridoxal phosphate. Walang pyridoxal phosphate. Meron mga reaction, right? So, so you have to remember, guys, pyridoxal phosphate is involved in what reactions? Di ba? Parang this is a high order thinking skill. This is a high order thinking skill question, right? Right, because you're not asking a direct recall or retention lang, no? It's analysis, right? Carboxylation, tell me. Ito na lang, no? Let's expand the question. Carboxylation, what is the coenzyme? Carboxylation, what is the coenzyme? I know you know the answers, right? Your answers are correct. Pero let's put some value into the question. Carboxylation, what is the coenzyme? Meron ba ako dito? Ayun, Biocytin, di ba? Biocytin. Carboxylation, di ba? Yung OC. Carboxylation. Hindi decarboxylation, ha? Carboxylation. Pag carboxylation is biocytin. Okay? Carboxylation is biocytin. Methylation. Methylation is one carbon, di ba? Methyl is one carbon group. Methylation. Methyl is a one carbon group. So what is involved with methylation? Anyone? Sige, I'll wait. Ay, by the way, if you haven't subscribed yet, shameless plug, no? If you haven't subscribed yet, kasi in a few na lang, I'll be reaching 1,000 subscribers. So magkalapit nga kayo sa mga dumadaan sa kalye. O subscribe naman kayo, kawawa naman si Doc Bang. Anyway, sige, going back. Sige, let's go look at your comments. Okay. So your methylation can either be what? Can either be, right, no? Your tetrahydrofolate, di ba? Formyl, methanol, methylene, methyl group, right? Or cobamide, no? Cobamide, yung methylcobalamin, right? Methylcobalamin. Hydroxylation, especially of your aromatic amino acids. Hydroxylation of your aromatic amino acids. Tama, JMS Bondok, right, no? Tetrahydrofolate. Tama. Pero yung letter D, hydroxylation. Tingnan mo, my point exactly, 'di ba, guys? Because there are so many, you have 11. Sana nga ba yun, alam ko yun, hydroxylation, eh. Hydroxylation, yes, it's tetrahydrobiopterin, right, Angel? So, Doc, alam ko may hydroxylation, eh. Ano nga ba yun? Tetrahydrobiopterin o tetrahydrofolate? Okay, so yun, no? So, dapat alamin din natin, dapat alamin. Tama, Thea, no? Hydroxylation, tetrahydrobiopterin. Okay? Sige. So, the answer here is A. Okay? So, ayan na naman siya, di ba? Parang sikat na sikat si pyridoxal phosphate. Baka nga dapat aralin si pyridoxal phosphate. Hindi, pero seriously, when you go to your amino acid metabolism, pyridoxal phosphate is very important. Okay, and, so question number 7. 100 questions to, guys. Hindi, just joking. Konti lang. Okay, methotrexate and sulfonamides are folic acid antagonists, right? They're antifolates, eh. Which of the following enzymes will be inhibited by this antagonist? So, high order thinking skill na naman yan. Hindi siya kasi yung direct question, eh. Right? So, what we're going to do here is, for each of the enzyme, let's try to match it with the different coenzymes. Acetyl coenzyme A carboxylase. Ano nga ba yung carboxylase? Carboxylation, addition of carbon dioxide. Addition of carbon dioxide is Biocytin, di ba? Biocytin. Malate dehydrogenase is what? No? Dehydrogenase is just a simple what? Dehydrogenase is just a simple oxidation reduction. Dehydrogenase, simple oxidation yan, eh. So, malate dehydrogenase, I believe, is NAD. NAD. Alanine aminotransferase. This is just a simple transamination. So, alanine aminotransferase or transamination is pyridoxal phosphate. So, we're left with serine hydroxymethyltransferase. Remember, serine to glycine. Serine to glycine, no, you remove your CH2OH, no, that's hydroxymethyl, right? But in this case, you what? No, you transfer your methyl, right? So it's you transfer your methyl. No, it carries your one carbon group. So the answer here is your serine hydroxymethyl transferase. But if there's a question, no, if there's a question, wala yung serine hydroxymethyl transferase, what's the other? What's the other answer? Is your thymidylate synthase. Thymidylate synthase. Thymidylate synthase. No? Synthesis of your thymine, right, from your DTMP, no, your deoxythymidylate monophosphate from your deoxyuridyl monophosphate. Okay. So, letter C. Carboxylation of pyruvate to form oxaloacetate is facilitated by which of the following coenzymes? Hala, hindi ko yata alam ang sagot dito. Okay, pero carboxylation, alam na natin yan, di ba? Formyl transferase. Formyl transferase. Teka, may tinatanong si Timothy. Formyl transferase is for tetrahydrofolate. Pwede rin, right. Tetrahydrofolate. Very good, right. Formyl transferase as well, right. Timothy, you're right. If you can see this is the tatlo siya, right? So, yan, Timothy, that's right as well, no? So you have 3, no? You have your serine hydroxymethyl transferase, your thymidylate synthase, and your formyl transferase. Very good. Okay, very good. Sige. Carboxylation of pyruvate to form oxaloacetate is facilitated by which of the following coenzymes? Coenzyme Q, definitely not, right? Because it's responsible of accepting electrons from complex 1 and complex 2. Flavin adenine dinucleotide, that is FAD. This redox rin lang eh, di ba? That is redox. Thiamine pyrophosphate. What is thiamine pyrophosphate again? What's thiamine pyrophosphate? Nakalimutan ko na rin. No? Thiamine pyrophosphate. That's involved in your alam ko na, in your non-oxidative decarboxylation, etc. Okay. So here, it's carboxylation. So it's your so the carboxylation, thiamine pyrophosphate, carboxylation. The ano pala dito, the explanation here is Letter B and letter C is definitely not, right? So you have to choose between biocytin and thiamine pyrophosphate. Thiamine pyrophosphate is involved in decarboxylation, no? Oxidative or non-oxidative decarboxylation. So the answer here is biocytin because it is carboxylation. Carboxylation. Okay, question 9. All of the following coenzymes participate in redox reaction except. Ito, low order lang to. Low order thinking skills lang yan. Right? What are the four redox coenzymes? What are the four redox coenzymes? You have your pyridine nucleotides, you have your flavin nucleotides, right? You have your ubiquinone, and you have your, kasi ditong lituhan yan eh, ditong ano yan, dito magkakatalo yan. Ano nga ba, tetrahydrobiopterin or tetrahydrofolate? Tetrahydrobiopterin, di ba, is what? Is one of the first four. Di ba, apat yan? NAD, FAD, ubiquinone, and your tetrahydrobiopterin, right? So tetrahydrofolate is not a redox, right? It is involved in the transfer of one carbon groups. Okay. So that's the answer. Mahirap ba ang coenzymes? Guys, coenzymes mo mahirap? Actually, they're simple, but the problem is they're overwhelming. Right? They have a lot. But just remember the coenzyme shelf, the info shelf that I put in the left side. In the left side, yung summary yan, that's the summarize. A lot to memorize. I know, Ian, no? I commensurate. I empathize. They're hard to memorize in one sitting, no? Unless you have a super mind, okay? But you do it one at a time. No, in Pomodoro, kunyari magpa-Pomodoro technique kayo ng 25 minutes and 5 minutes, siguro one group, you study one group. Pero for me, what I really recommend, you separate them, you study them in different days. Para you can associate them with certain, alam ko yan, I was in the living room, alam ko yan, yung tetrahydrobiopterin, una ko yung napag-aralan, nasa living room ako yan bago mag-dinner. Ganun, that will help you. Otherwise, if you're going to study them all at the same time, wala labo-labo, rambulan yan, ready to rumble in your mind. Okay, it's hard. Saka you have to, again, read and read. Okay. Question 10. In megaloblastic anemia secondary to vitamin B12 deficiency, which reaction will be inhibited? So it's very important to really remember the different reactions. Okay, guys? No? So it's really important to remember the different reactions. Pyruvate dehydrogenase complex, you don't see vitamin B12, right? You don't see vitamin B12 in your pyruvate dehydrogenase complex. You only see 5, di ba? The 5 is your thiamine pyrophosphate, lipoamide, coenzyme A, FAD, NAD. So letter A is not. Aspartate transaminase, for aspartate transaminase, what is the coenzyme? For aspartate transaminase, what is the coenzyme? Aspartate transaminase, the coenzyme is your, di ba, transamination of amino acids, right? It's your pyridoxal phosphate, tama iyan, no? So, hindi rin pwedeng letter C. Pyruvate carboxylase is biocytin, right? Carboxylase is biocytin, right? So, here, the answer here is your methionine synthase, methionine synthase. Kasi it's vitamin B12, it's methylcobalamin. Please do remember, methylcobalamin for methionine synthase, dalawa lang naman sila eh. Then your what's the malonyl coenzyme A, methyl malonyl coenzyme A mutase, that is your adenosylcobalamin. That's your adenosylcobalamin. So methionine synthase. Okay. Question 11. Coenzyme A synthesized from which of the following vitamins? Yan, simple lang yan. Answer? Niacin is niacin. Niacin, vitamin B what? Niacin, vitamin B. Which vitamin B gives rise to NAD? Which vitamin B gives rise to NAD? B3, tama, di ba? Niacin. 3 syllables. Niacin, 3 syllables. So vitamin B3. Riboflavin is vitamin B2. B7, B7, 7 biotin, 7 biotin, right? So coenzyme A is, yan, no? Sa NAD, NADP, FAD, FMN, vitamin B7 is biocytin, 7. Biocytin, 7. O, di ba? Alam na ni Angel. No, biocytin, 7. Pantothenate, why? Because it's CoASH. CoASH, 5 letters. Vitamin B5, 5. Pentatonics, penta. So it's pantothenic acid, which is vitamin B5. Right? I know it's corny, no, but hopefully, no, sana makatulong. Okay. Q12, question 12. Which of the following coenzymes is derived from vitamin B9? Ala, ano nga ba yung B9? Bilangin mo yung letters, no? F-O-L-I-C, A-C-I-D. Okay. So, niacin, vitamin B3, thiamine pyrophosphate is vitamin B1, Flavin mononucleotide is riboflavin, vitamin B2. Your tetrahydrofolate is vitamin B, what, no? Vitamin B9. Folic acid, 9 letters. Folic acid, F-O-L-I-C, ACID, vitamin B9. Okay, vitamin B9, tetrahydrofolate. Okay, tetrahydrofolic acid. Very good, Mec. Tama. Okay, Angel. Tetrahydrofolic acid. Mec, tama. Okay. If an individual has vitamin B6 deficiency, which of the following amino acids can still be synthesized and be considered non-essential? Allah. Sige nga, hulaan natin. If an individual has a vitamin B6 deficiency, which of the following amino acids can still be synthesized and be considered non-essential? Wala. And silence pervades the live chat room. Hindi, madali lang yan, guys. Madali lang yan. Ang galing. Tama, Ian. Tama, Teya. Ian, Teya, you got it right. Mec, Prad, tama, right? Serine, alanine. Alanine and aspartate, alam natin yan. Alanine and aspartate. Serine, alanine, aspartate. What are the essential amino acids nga pala? So all of this non-essential, right? Tyrosine, serine, alanine, aspartate are all non-essential, di ba? Private team hall. Yung T naman dun is, ang T dun is tryptophan, right? Di ba? Tapos M, S, alanine, aspartate. So lahat non-essential, right? Pero if vitamin B6 is deficient, what happens? Alanine and aspartate, no, which you can synthesize from transamination of pyruvate and oxaloacetate. Di ba yung ALT, AST? Right? So, hindi pwede, right? Kasi pyridoxal phosphate is responsible for what? B6, pyridoxal phosphate, right? What are the reactions facilitated by pyridoxal phosphate? Amino acids yan, right? Di ba? Transamination, di ba? Decarboxylation, etc. Serine, alanine, and aspartate can be synthesized via transamination, right? So all of them will not be synthesized. Will not be synthesized with vitamin B6 deficiency. Tyrosine, where do we get tyrosine? Guys, where is tyrosine derived from? Di ba tyrosine, just like phenylalanine, is an aromatic amino acid? Right? Aromatic amino acid. Right, Ian, no? Phenylalanine. John Lester Fabella, right, bro, right, no? It's phenylalanine, right? So how do you convert phenylalanine to tyrosine? Di ba? You have your aromatic amino acid hydroxylase, right? So phenylalanine is converted to your tyrosine, but you don't use your transamination. You don't need pyridoxal phosphate. Very good. Right, Thea. Phenylalanine hydroxylase, you need your tetrahydrobiopterin. A tetrahydrobiopterin is not vitamin source, right? It is independent of vitamin sources. So the answer here is, tama lahat, no? Tama kayong Mec Angel. No, it is your tyrosine. Tyrosine is DEDMA to your pyridoxal phosphate. No, tyrosine is not dependent on pyridoxal phosphate as most of your amino acids are. Did you get that? Do you have any questions? Did you get that? Tyrosine is a league of its own, so it's above the others, right? Kasi most of these, no, when you're going to study your amino acid metabolism, most of these are what? Are synthesized via transamination. Okay, I hope you got that, huh? Question 14. By the way, 100 questions. So no, I'm just joking. All of the following coenzymes are examples of prosthetic groups. Ayun, what do we mean by prosthetic groups? Prosthetic groups, the coenzymes are attached tightly. Tightly. They don't leave. They're always attached to your apoenzyme through a lysine-amide linkage. What do you think is the answer? We went through this kanina. I explained through this. I went to the great lengths to explain this. No, I explained why they are bound, no, and how they are bound. Mostly they are bound by an amide linkage to the epsilon amine of lysine. Right. JMS Bondok, Ian Cedric Thea, you're right. Biocytin, di ba yung biocytin, biotin, di ba? Then you have a component and then lysine, etc. Same with pyridoxal phosphate and lipoic acid. Kaya nga siya lipoamide, right? Lipoamide siya, hindi siya lipoic acid. Lipoic acid does not exist, no? It's lipoamide. Ubiquinone is mobile, is hydrophobic, di ba? It can make sundo the electrons from your NAD and from your FAD and bring it to your complex 3. So the answer here is what? No, it's ubiquinone. It's not bound. It's mobile. So the answer here is ubiquinone. Question 15. Sikat si pyridoxal phosphate. No? Pyridoxal phosphate is required for which of the following reaction pathways or individual reactions? Phenylalanine to tyrosine, we just discussed that. No? That is tetrahydrobiopterin, right? Right? Propionyl CoA to succinyl CoA, it's a rearrangement. No, it's rearrangement via methylmalonyl CoA. Methylmalonyl CoA mutase, right? So that is your Cobamide. Then pyruvate to acetyl CoA, we know that. That's your pyruvate dehydrogenase complex, right? So it's clear. The answer is clear, right? Aspartate and alpha ketoglutarate. Now, this is your amino acid. You take the amino acid here, the amino group, you add it to your alpha keto acid, no, alpha ketoglutaric acid. What happens? Aspartate becomes oxaloacetate. Your alpha ketoglutarate will become glutamate, and that is transamination. That is what? No, that is facilitated by your pyridoxal phosphate. So that is tetrahydrobiopterin. That is cobamide. And that is your pyruvate dehydrogenase complex, oxidative decarboxylation. And tada, the answer is letter B. Most of you got it, no? Shout out to Timothy Matthew, David Christian, Angel, Ian Cedric, Thea, right. Okay, and that is the last question. So thank you very much again, So 2 hours and 22 minutes, I apologize. So anyway, there are 11, right? So if you're 120 minutes divided by 11, so around ano lang, no? So around 10 minutes lang, tama ba? Tama ba 'yun? 10 minutes lang per may questions pa tayo, no? So I think it's good. It's a good time. Do you have any questions? So to everyone, no, to the 33,000 viewers, 30,000 viewers, thank you very much for staying up this late, Staying up this late and listening. I hope I was able to impart something helpful. So thank you very much, Timothy, David, Angel, Ian, Thea, to everyone. Okay, to MacProd, Timothy, Matthew, John, Lester, Fabilla, Ian, Cedric. So thank you very much. Ayan, no? Thea, sorry. Thank you then Ian. Thank you, Rodora. Oh. Itong gusto ko, itong quote of the night. When in doubt, always pyridoxal phosphate. Thea Gutay, 2021. Michaela, you're welcome. Bro, you're welcome, bro. Kung may questions kayo ha. So, by the way, wala pa kasi akong community tab eh. If you have any requests, kasi kundi magpapapoll ako eh. Just I need a few more subscribers. Then once I reach 1,000 subscribers, I can post na. No? I can have a community tab on YouTube. Okay, so ang shameless plug niyan, please subscribe. So, thank you Len Angel. Stay safe, Len Timothy. Ha? Stay safe. Okay, no questions. No questions. Para ako may echolalia, no? I always keep on repeating. Wala, that's my ano. David Christian. Paano kami? David Christian Calma. Okay. Thank you very much. So, thank you guys. So, Mercozy, that's a new name, ha? That's a new name. So, welcome. Ayan, thank you, Mek. Okay, sige. So, I'll send you my check. I'll send you. I-Gcash ko na lang. Thank you, Mek. Hindi, you just share. Ask them to subscribe and share the video. I do admit, nahirapan din ako mag-prepare eh, kasi you have 11. If there are any errors, you point it out, ha? Kasi I'm sure I did commit some errors. I have never lectured this in the past, no? So I prepared for this lecture, no? It was not, I haven't lectured this in my 25 years of teaching. Anyway, but I'm also learning din naman eh. So sabay-sabay tayo natututo. Any questions? I will rewatch your videos. Thank you, Mer. Thank you. Welcome. Naka-ano ka na, type B. Ayan, o sige. Tristan, thank you for, dati sobrang lito ako at least now medyo. Basta you study them by groups. You study them by groups and don't study them at the same time. No. Road to community tab, yes. Pero by October 12 daw they will bring it down to 500 subscribers. Anyway, okay. So thank you guys. So always stay safe. So good luck on your exams. I know you have a lot of exams next week, and next week it's shifting exams. No, so I'll include you in my prayers every night. So laban lang ha. First year is always the hardest because it's basic. No, you're still adjusting. But what will not kill you will only make you stronger. So you're building strength upon strength. Strength upon strength. Of course, I'll not forget you. Don't forget me as well. Okay. Anyway, so good luck on your exams. So, David Christian Calma. Useful for the books. Kaya nga pala familiar yung name mo eh, David. What? David Christian Calma. Okay. So, kaya nga pa I know you. Sige. I'll end the broadcast. Thank you very much again. And if you have any requests, you just comment. If you have any requests for me for any live stream, it will be my pleasure. Thiamine inactivated by Suna, di ba? Thiazole group. Gusto ko yung ano eh, when in doubt, kanino eh, when in doubt, Pyridoxal Phosphate, Thea Gutay, 2021. Okay. Sige, night guys. Sige, ganyan na lang. I'll end the broadcast. Batch 2020, kaya sige. Welcome, David, ha. So, good luck on the boards, ha. Bye-bye. Bye-bye.